Yeah, Ankur, please.
Yeah, yeah. So, good morning, all. First, I will present the session timeline, like how session is goes. Is my screen visible?
Not yet. Yeah, that's coming. Yeah.
Yeah. So, in the first session, welcome and introduction to SilicoScientia by Dr. M. Atul Islam. Then, in the second session, there is a keynote lecture on high throughput virtual screening with machine learning by Vikram Suryavanshi, junior computer chemist in SilicoScientia. Then from 11 to 1:45, 11 a.m. to 1:45 p.m. hands-on training
Yeah, Ankur,
by
please.
whole SilicoScientia team, where you have divided already divided in the groups.
Good
You will
morning,
get
all.
the link, and you have to
First,
get the
I will present
hands-on
the
training. And after the
session.
completion of training, there is a conclusion on from 1:45 to 2 p.m. Yeah. So, good morning. On behalf of SilicoScientia, it gives me immense pleasure to welcome you all on the to the hands-on workshop on AI in drug discovery. As the program coordinator, I am proud to host this significant scientific gathering that brings together a global audience to explore the transformative power of artificial intelligence in pharmaceutical research and development. This workshop is a part of our commitment to bridging computational innovation and biological insights, providing researchers, students, and professionals a practical and enriching learning experience. We are privileged to begin our workshop with the welcome and address by Dr. M. Islam, an internationally recognized expert in computational drug discovery and the visionary founder of SilicoScientia. Dr. Islam holds a PhD in pharmaceutical and fine chemical technology from the University of Calcutta and brings over 15 years of extensive global research experience. His distinguished career includes prestigious fellowships such as Newton International Fellowship UK and the NRA Postdoctoral Fellowship South Africa. Prior to founding SilicoScientia, Dr. Islam served as a senior computational chemist at 3 Bigs Omiscore, Bangalore, and held academics and research roles at the University of Manchester and the University of Pretoria. A prolific researcher with more than 50 high-impact peer-review publications, Dr. Islam work spans QSR modeling, molecular docking, pharmacophore mapping, AI-driven virtual screening, molecular dynamics, and inhibitor designing for emerging and neglected diseases. This session will provide a comprehensive overview of SilicoScientia missions, innovation, and roadmap, highlighting our dedication to fostering a collaborative, inclusive ecosystem for scientific advancement in AI and drug discovery. Thank you. Atul sir. Over to you.
Okay. Thank you, Omkar. And once again, on behalf of SilicoScientia, I'm welcoming all of you from across the globe. And this is our second workshop. So I think a lot of people also here, those already attended our first workshop. So as Omkar mentioned, this is actually the objective of the workshop to give the insight how can we use the artificial intelligence method discovery. So I think the details about today's theme we presented after my presentation, but I would like to mention that is the artificial intelligence. Actually, it is a powerful approach or powerful methods, and that you can use in the field of drug discovery. So the last time we have conducted a workshop on how we can use the artificial intelligence in the docking. So this time actually a little bit different. So here we'd like to go for how can we screening the large chemical database. So we would like to develop the model from the unknown data set, and then that data set can be used to screen the larger chemical space, and followed by the traditional docking and the pharmacokinetics and how can I get the final output. So these are the series of workshop is conducting by the SilicoScientia, So step by step, because in a one day or a half day, it is not possible to cover up all those things. So that is this type of workshop will come frequently to cover up the different topics. Okay. So I would like to explain about the company's mission, company's vision, and what exactly we are doing and why we are doing these things. So that actually I took to explain before you, and then we'll go for the technical session. Okay. So my screen is visible, am I right?
Yes, sir.
Yes, sir.
So I think those people already attended our last session, or maybe some. I saw that is some students from SSR college also are there. So I presented my presentation in last this week only. The SSR students. So I think the SilicoScientia started 2 years back in Bangalore. So our reason to apply that computational biology and computational chemistry to transform the healthcare sector, because if you see during the evolution of the computational resources, computational power, the people are eager to introduce the different approaches. So similarly, the computational chemists, computational biologists, they are applying the beauty or the power, the beauty, the strength of the artificial intelligence. So that is our vision. That is, yes, how can we change the healthcare sector in regards to the disease management, in regards to the uh, potential molecule development, in regards to the management of the healthcare data. So that is our long-term goal. And our mission, that is, we are working in a different, diverse kind of diseases. So we're trying to find out the potential molecule for the diseases. Along with that, how can we give the easy to use solution to the non-technical person, like those people are not very well sound in computers, like especially Linux or the Python programming or any kind of programming. Because people are, some people that are, especially the scientists or the people from non-IT background, they're scared about the programming language. So how can we give the uh, easy to use solution for the complete drug discovery and the bioinformatics? This is our mission. So we have developed our own platform that actually I'll briefly explain in the later stage of my presentation. So the SilicoScientia, I mentioned, a Bangalore-based company, and we are already in startup India and also MSME. And whatever the product we are developing, it is this product Make India product, because it is our in-house developed by ourselves. So it is definitely the Make India scheme. So what actually we do, we combine the bioinformatics, we combine the cheminformatics, and also we combine the uh, effectiveness or the power of the artificial intelligence. And here we also engaging with the industrial partners, academic partners, and research organizations. So different organizations, a lot of people from academic background here, a lot of people from the industry background here. So you know the different org culture, uh, different way the engagement are there. So we're trying to uh, engaging with the academic collaboration as well as the research organization as well as the industry partner to help or to get the more insight about this computational drug discovery, and definitely building the solutions of this in cycles of taste, learn, improve, and update. So this is our main motto, that is to work, test, and learn from there and improve it. And definitely, we are refining our approaches day by day. Definitely, the real time applications are there, because ultimately we have to go to the uh, real system. So these are the things actually currently the SilicoScientia is offering, like uh, sorry, like computational drug discovery, that is the huge computational chemistry. We use the traditional computational drug discovery approach. We use this artificial intelligence, machine learning. Also, we offer the bioinformatics solution, and definitely the artificial intelligence, how we can use in the healthcare sector. Apart from that, also we provide the training program, that is how to skill development of the people from the academic background, maybe the faculty, maybe the researchers, maybe the scientists, or maybe the students, or maybe from the industry setup. Also, the people may come to us to develop their skills. So we provide according to their need. We provide the training to the people. Also, we provide give support to the academic researchers, academic research support. Like if we have the problem, come to us. We'll give the solution. We make the research problem. We identify the research problem, then we develop the pipeline and execute it. So that also we provided. So actually, I was one of the co-founders and directors of the company, and actually, in my last long journey in this field, I worked in a different academic and corporate setup, and I found what are the limitations are there, and how can we provide easiness to the people. Because honestly speaking, I talked to the different people, like from the different organization, and also I found that the people can manage the limited data, but when the data are expanding, people are scared to work on that. So how can we actually this thing? Because in a long time, when the different people are actually coming to us and giving this, yes, this is the problem. How can manage this? This is the data. I have talked this kind of molecules. I spin this kind of molecule, but I am able to understand how to finalize the molecule, how to what are the parameters I should take. So these things actually came in mind, and then we said, so why we are not giving some solution, that the people should easily understand and easily can work in this field. So along with me, Dr. Sonali Thought, she's one of the co-founders and directors, and she has also a long journey in this field. She's basically that is a synthetic chemist. She has experience in India as well as abroad. Uh, so all these things actually will come together and we discuss this, and that actually we have decided to do something to make the things easier for the people. So this is a team actually, the Bioinformatics team we have. That is, they are from the bioinformatics background, they're from the pharmaceutical background, they're from chemistry background, and they are expertise in a different field, and now they are working in the computational drug discovery. We have our IT team, that is, we are actually working to develop the platforms. So already we have developed our computational drug discovery platform. So some people from the bioinformatics background, some people from the IT, and I'll show you some slides of our platform. Also, we have the bioinformatics, similar to the computational drug discovery. We are developing our own platform, the bioinformatics. We already developed some uh, pipelines, and also definitely this AI machine learning team. They are in the pioneer, and they are helping to all this IT or informatics or bioinformatics team. How can we use the machine learning approaches to make the things much better, much efficient. So their role in here in SilicoScientia. So as I mentioned, that is the long journey I understood. That is, the people are struggling, the molecule is struggling in this field. How can we accelerate the drug discovery process, and how, due to the resource, the accuracy, we need to compromise the accuracy. And definitely, if you don't have the good infrastructure, it is taking the time, and it is definitely an iterative process. So in this situation, uh, we thought that is, as I mentioned, that is, we should provide some smart technique, and that is the objective of the SilicoScientia. So this is not the full solutions or full approaches, but we do the identification of target to validate the biological, uh, whether the target is the biological references, and definitely in the iterative process, we improve. We are improving our prediction, and also we optimize our streamline, integrate the feedback loops, and then definitely, as I mentioned, we are in collaboration with the different organizations, the academic, non-academic organizations who are collaborative, and try to make a good ecosystem. But from our services in the different field, we have our own R&D. So we are working in the different R&D project, and the last 2 years, more than 15 research articles we have published already in the different high impact journals. So few of them are here. That is how we can use machine learning, and also how can we use the physics based method to get some potentiality in this field and get some potential molecules. So this is our journey. Actually, we started in 2023, and then we put on the first project from the University of King South in March 2024. In September, we have started our work with the Symbiosis International University in Pune. In the same month, in September 2024, we are also engaged with another Department of Pharmacy in South University. Then in October 2024, we are collaborated with one of the industrial partners in Bangalore, Decodage. We work with them also. Then another university from the Finland, Warsaw, that is Finland, that in December 2024, we have our own that is peptide work. That is, we are designing the peptide. The validation is going on in that university. And in March 2025, we are started the collaboration with the DI Patil Pune. So we have launched one year diploma course. So Vikram will talk about this course, that is what is the objective of the course and how it is executing. And also in March 2025, we have launched our product, that is Silico Explore, that is a comprehensive computational drug discovery platform. So that already the license given to Nagpur University. And also we have the collaboration in March 2025 with the CSIR, CDRI in Lucknow. So these are the brief about our journey. So apart from that, a lot of other achievements are there, like publications are there, some faculty development program is going on. So these are the brief about our journey. So what I was talking about our product, that is what we have developed. So Silico Explore, it is a cloud-based machine learning assisted physics-based computational drug discovery platform. So here till now, more than 25 tools are there. So few are third-party tools we have integrated here, So in the integration, the main purpose of the integration to make it easy to use and handle the large number of data. And after the execution, the analysis, the post analysis, the post performance analysis that we have implemented, that will help the user to take the decision. So, along with the docking, we have our modeling tool, we have our toxicity prediction tool, the confirmation injection tool, the physical chemical parameters analysis tool, we have the interaction, binding interaction, and also you can identify the target. You have the deal the large number of molecules, how can we convert to each other? And definitely the one of the biggest, one of the beautiful tool in the molecular operation targets for the molecular dynamic simulation that also we have indicated here. We have also the mutation inducer, that is what is the impact of the mutation in your protein, pharmacokinetics, then machine learning based screening. So today we'll show you some glimpses of that. Because this is due to the time constraint, we can't go in a detail, but we can give you the idea of how we can use the machine learning to screen the large molecule. So in a robust models are already integrated in our platform. And we have the similarity searching from using non-molecule. We have the genome generator that you can generate new molecules from your standard molecule. You can generate a peptide, that is I told already, that is our collaboration with Finland University. Rescoring and the pharmacokinetics. So these are actually the tools, and it is still the more tools are coming in the next 2-3 months. So these are actually already I mentioned. These are the tools, like the docking tools are here. So all the 4 docking tools we have integrated with the different algorithms are there. There different approaches are there, like ligand or Bina. These are the more or less similar. The difference is the algorithm, also the hand of the different algorithm. For the diffuse bind, that is a more or less validation of the docking tool, docking data, how can I validate. So here are some prediction tools for the pharmacokinetics, that is physical chemical toxicity. These are some analytical tools. This is some visualization tools. Apart from that, I already mentioned that we have the conversion tool, we have the dynamics tools, the mutation inducer tools. So these are the tools are right now. Apart from that, as I mentioned, the more tools will come, like when proposing the new molecular docking tool developed by SilicoScientia. Also, how can we calculate the age depending on the mutation, the crystal structure prediction of the small molecule, that also are coming very soon. The SAR already integrated, and also I said that we are developing our independent bioinformatics platform. So the RNA-seq is one of the part of that. So this already I think I mentioned, and I'll show you later, that is already we have some scalable success, like we have given the internship around more than 30 to 35 people across the globe. Some people from South Africa also. We have trained them in a different field, cheminformatics or bioinformatics or the machine learning. We also we had interns from the management background. We already launched the course in artificial intelligence in computer drug discovery. And also I mentioned that we have already launched the Silico Explorer, which is our exclusive product of SilicoScientia. And another the milestones very soon will come, that is we're going to launch the course in MSc in Bioinformatics, maybe coming here with some one of the universities in India. So these are our the partners or collaborators or clients. So Kingston University I mentioned, Bharati Vidyapeeth, then that is the International Institute of Molecular and Cell Biology, the Finland, and DI Patil, Symbiosis University of Calcutta. Picardo is our industry partner, and definitely the CDRI. So these are actually our the market that is we are working in the with the different pharmaceutical companies. We're working in the research institution. We're also working in the model of contract research organization, and definitely the government and non-profit research agency. So we are diversification ourselves across the globe uh, to work with the different people. So our model that is B2B, B2C, and B2G, that is business to business, business to client, and business to government also. So this already I mentioned that our main focus on the SaaS licensing, that is our platform, zero services, the academic courses that is already I mentioned, training programming, and also supporting the research in the different organization. So this is also already I mentioned about this. That is what actually we have achieved. So along with that, we have also published a significant number of articles, the different field and the different high impact journal. So overall, we are actually in the domain of the computational chemistry and computational biology to work in a diverse domain. So and also to train the people, those people actually they heard about what is the artificial intelligence, but they don't know how to implement artificial intelligence in the computational discovery process. So yeah, that's all from my side. So thank you so much for joining in this workshop, and I hope we'll have a good time with us, and you will learn something from here, and then that you can use your own research. Thank you so much. If you have any questions, you are welcome to ask. Otherwise, we will move to the next session.
If anyone has any question,
If you have any question, just you can unmute yourself and ask.
or you can add in chat your question. You can unmute yourself, or you can just type in your chat and say it.
Okay. Thank you, Amkar. You can proceed.
Okay, so no one has any question. So we will move to the okay. So can you help me in drafting my MSc proposal including ML?
Yeah, sure, definitely. Yeah. Uh, Krishna, we can help. We need to talk to you. What is your interest, and what if you have any predefined data or predefined disease model, you can draft the pipeline, or maybe if you don't have, then we'll give you some suggestion. So definitely we can do. Keep in touch with us. After session in the talk, or anytime you can contact, we'll definitely help you. Yeah.
Thank you so much, sir.
In the second session, there is lecture on high throughput water screening with machine learning by Vikram Suryavanshi. Vikram is a junior computational chemist in SilicoScientia. Also, he has completed a master's in pharmaceutical chemistry. So we are excited to present a keynote lecture by Vikram Suryavanshi on the high throughput water screening with machine learning. In this session, Vikram will delve into the integration of machine learning techniques in accelerating the virtual screening workflow. This lecture will cover the key concept of machine learning and their algorithm, and how it works, and how we will actually use in the screening part, all this in the drug discovery pipelines. So thank you, Vikram. Over to you.
Okay. Thank you, Omkar.
Okay, so is it my screen is visible?
Yes.
Okay, so good morning everyone and welcome you all in this hands-on practice workshop in the AI based drug discovery. So in this session, I am going to introduce and guide about the high throughput virtual screening with machine learning. So how this virtual screening works in the machine learning methods and how we can get the screen the high number of databases into the smaller number as a potential molecule. So from this session, I am going to introduce all the steps and ML-based execution regarding the this virtual screening. So let us begin. So first, what is drug discovery and development? You all are aware about the drug discovery process. So it is a very lengthy process, and it is generally based on different types of targets and molecules. So it contains various type of steps, starting from the pre-drug discovery, then target identification, lead identification, its optimization, and clinical trials, and lastly post-marketing surveillance. So it is a very lengthy process, is about 10 to 15 years. So as quick review, drug discovery is a process as identifying new potential drug molecule from the initial study or any type of data available. So this drug discovery is very challenging nowadays. So in particular sector, we are going to introduce AI-based and ML-based modeling, learning-based mechanism for the easy and quick
Yeah,
execution of the process.
Vikram, I think your slides are not moving.
Yes, yes, sir. Is
Yeah,
it moving, sir?
yes.
Yes, okay, sir. So what is drug discovery? So drug discovery contains so many types of different approaches for the process, like starting from target identification. The target identification means we are going to study the deep info about our target, its mechanism and all. Then the virtual screening, large number of data visits, we are going to virtual use screen using the various computational methods like molecular modeling, then the dynamic simulation, then docking simulation. So virtual screening is also the approach for the drug discovery. Then drug repurposing is also term comes in under the drug discovery process. So many types of drugs are repurposed as another medications for the treatment. Like as an example, uh, as a antihypertensive drug in the market, diuretics has comes as a both the medications like antihypertensive and it's a diuretics mechanism. So we can also do the various type of research as a drug repurposing as a drug discovery. So in the drug repurposing also we can add ML models and ML-based algorithm for prediction of its activity for the different types of the protein targeted and its inhibitor action. Then toxicity prediction and structural activity relationship study. So we can also predict the toxicity of the compound according to their structural feature available in the molecule, like its pharmacophoric features and pharmacophore oxochrome available in the functional group and related to toxicity like sulfur. Sometimes sulfur is very toxic atom in the compound. So we can also detect and using the AIML based model and we can predict the toxicity. So this field is also very emerging, like toxicity prediction and SAR point of view. SAR is a very wild field in the medicinal chemistry part. According to structural changes and modification, we can modify the drug molecules activity. So next, the de novo drug designing. De novo drug designing is also a process in which by using the existing compound, we are going to design novel compound using the same compounds by various approaches like its structural modification or its biological activity detection or mechanism study and all. And last is the optimization of the final drug molecule. So this is a basic workflow and steps in the drug discovery processes.
And apart from that, as a workflow, it contains so many processes like starting for target identification to up to the post-marketing surveillance. So these are the very lengthy process. So using this ML-based integration and artificial intelligence as a quick and rapid, we can create the model and train the model and give the result and predict the compound activity or any other data prediction as in the rapid time and less cost. So it is a beneficial here in the drug discovery, this field AI ML based algorithm field. Then what are the challenges in drug discovery? So there are so many challenges. First is it required stable financial stability and high cost because starting from the hit identification to last clinical trials and all, it required so many types of funds and all. So it is high cost and it is also time consuming and it also the low success rate because we are going to start with more molecules like in lakh or billion compound, but lastly there is low chances we got the final compound as a this means potential compound we got or not. If we got how many number we got, is it in the good number or bad number? So the low success rate is also here the experimental validation. So to overcome these lengthy experimental validations and this high cost and time, this artificial intelligence and the machine learning method is so much easier and the best option for this current drug discovery approaches. Then come to the point where today we will go for the hands-on training. So today we are going to learn about high throughput virtual screening using the ML-based model and screening of the large database in the quick time and less high throughput virtual screening. So it is a very effective and time-saving method. You can see here it contains large numbers of molecules as a starting of this step. So at starting we are going to take a large number of compound and at the end of we can discover and find out the potential compound which are particular inhibitor of particular target. So this is a virtual screening. We are going to take a larger database and after the execution and all the research processes, it becomes the narrow down to a small number of candidates. So it is a very rapidly identified potential drug compound from the vast library of the compound. And in these processes, in the following step, we can do the molecular docking and simulations and various type of the computational chemistry approaches for the narrow down and for the identifying the potential compound from it. So so just in the short term, it is a narrow down the compounds from the larger bigger data set to the smaller data set using the various types of computational method. So in this workshop, we generated the ML based model and we trained the model. And according to that model, we taken the large library and lastly we created and we identify the potential compound from it. And we did the molecular docking and the pharmacokinetic analysis of this compound. So this is about the STVS process. So now this general workflow for our today's hands-on training on this session. So first is the machine learning for for compound activity prediction. And here we are going to train the model and build the model first and train the models. And then according to accuracy of the model, we are going to predict the activity of the new set of the compound. And from that activity, we are going to filter the compound according to their pharmacokinetic analysis, admit properties. And as a good compound, we are going to further carry forward the top compound for the molecular docking using Autodock Vina. And then we are also going to study the protein ligand interaction using the PLIP tool, protein ligand profiler. This is general basic workflow. Now, in this our approach, we utilize the random forest classifier algorithm for the development and building the model. So in the machine learning model, there are so many types of models are required and available for the building a model. Like SVM method is also available, then K-square mean, then various type of clustering processes are also involved. So this random forest classifier is generally based on the decision trees, and these trees are making the predictions. So each trees are the unique and unique features and all. So in this model, we constructed multiple decision tree and trained during this random forest classifier mechanism. So according to molecular features, each trees are converted into the features, and these features then converted into the outcome of the decision as a branch of the tree represented, and the each leaf node represented the final prediction. So from the different type of predictions and different types of the these features, as a final result, as concluded as based on the voting mechanism, like higher trees given the more result, and the 2 or 3 only giving the low result. So we can go for the higher side because it is we can go for the voting mechanism like model. So this is just introduction about random forest classifier we implemented in our workshop machine learning model. Then now the steps for the generation of model and the model training. So first prepare the data set for it and build a model. So you can collect the data set from any target specific data set from like binding DB is available, database, then KMDB is available, KMBL is available. So you can take a data set as a particular your target specific, and then you can model your you can create a training set according to their biological activity of the molecule, and label as a compound as a active or inactive. If the compound is active, so you can label as 1, and if the compound as inactive, you can label as 0. And then you can give this training data set to the pre-label model, and then with the help of ML training and random forest classifier, the smiles of the compound is directly converted into the Morgan fingerprints. Morgan fingerprint is a binary fingerprint in the numerical feature. It captures the structural and chemical features of the smiles and a compound, and it also give the properties and like which groups are absent in the structure on present in the structure, and it give finally the binary format of the Morgan fingerprint. And then we train the model using the random forest classifier, and then we evaluate the model score like accuracy and its RMS score and all. And then so we developed this model for the virtual screening, and then now next fact is we have the new compounds. Okay, new compound is means any compound inhibitor compound which we have to require the predict the activity of the biological activity of this compound. So we taken the compound from any natural compound databases, and made one test compound containing the smiles of that molecules. Okay, so in the screening of new compound on the trained model, we first load our random forest generated model, then we provide the data set of that new data set that we have to screen, and same the our model work. It featurized the smiles and analyzed smiles and converted into the Morgan fingerprint, and according to features, it labels the molecule as active and inactive molecules, 1 for active and 0 for inactive. So we got all the molecules biological activity after the last testing of the data, and we also provided option as a filtering of this active compounds. So lastly, it will filter the active molecules only as a result form, and it will give you the file format containing only the active compounds. Okay, suppose I am giving suppose 3000 compound to this model for prediction. So out of 3000, it will give around according to their features and all, it will give around 2800 or 2600 as active compounds. Okay, so from this active compound, we are going to next study their next algorithm, next mechanism. So this is about the machine learning based the building training and the testing of our model and the creation and identify the compound as active compounds. Okay, so next step is ADMET analysis. So what is ADMET? You are all aware about how this ADMET property is very essential for the drug discovery process. Any new drug molecule which comes in the market, it is very need to safe and non-toxic in the field. So here we implemented ADMET AI tool for this virtual screening process, and this ADMET AI tool works from deep learning algorithm platform, and it gives particularly 22 key properties of the ADMET properties, along with the toxicity also. So it is very useful to calculate the all features for the drug discovery and for the further computational works and for the synthesis of the molecule in the experimental validation purpose. So, from the train machine learning model and the describe and identify active compounds, those particular active compounds we are going to carry forward further for the ADMET study. Okay. So these are all the steps we are going to show you in the hands-on training by one. So you will get the knowledge about it. Then the general workflow for this ADMET calculation is we need the smileys for it, and then we have to give the smileys to this ADMET AI. Then it will calculate all the pharmacokinetic properties with safety and their efficacy scores along with the toxicity. Then according to their threshold and values like good or bad, is it good in the gastrointestinal permeability or in the BBB or something. So all the ADMET properties we need to check and select the good molecules for the further study. So we integrated the same filtration criteria in our generated flow. So you can train about this. Next is molecular docking. So we have to do the molecular docking of the same good potential compound which is we collected from the virtual screening. So first, what is the molecular docking? So molecular docking is a process where we can predict and identify the potential interaction between the protein and the ligand using the protein cavity. Okay. So what types of interaction they are showing with this protein cavity, active cavities and all. So we can do the molecular docking using two types. First is a rigid docking, second is a flexible docking. In rigid docking, generally both are in the rigid bodies like protein and your ligand molecules. And flexible, in the flexible docking, we are allowed to change the conformation of ligand or the protein molecule during the docking simulation. So by the traditional approach and traditional method using Autodoc Vina, this molecular docking takes so many time and steps for the execution. Generally, it takes first these four steps like first protein preparation, then ligand preparation, then generation of grid box on the active side of the protein and give the dimensions to the Autodoc Vina for the docking purpose, and the submission of docking to the vina.txt as a command line. So it is a very lengthy process as we are doing to traditional, but using ML based approaches and ML based model, we can automate this whole step in quick and within the short time period we can perform this molecular docking in our coming session. So as you seen in our last workshop, we already introduced the traditional methods of this molecular docking using Autodoc Vina. So we compare about these traditional methods and our ML based method, how they are very quick and easy to execution. So now how we implemented the molecular docking in our today's workshop. So first, from the starting ML based training and all model generation, then ADMET analysis. From the ADMET, we collected the potential compound, and that particular compounds we generate the ligand as a PDB format using automated script and model. And then we perform the batch molecular docking. Whatever numbers of compound we gather, like 1,000, 2,000 out of many compounds, we can easily perform the batch molecular docking with Autodoc Vina. And lastly, we are going to study the automated protein ligand interaction analysis using PLIP, protein ligand interaction profiler tool. Okay, it gives a detailed info about all the interaction between protein ligand, like hydrogen bonds interaction, pi interaction, pi cation, then Stahl bridges and all. So then lastly, overall session, we are going to show the interactive filtration and the result visualization of docking, and also the PLIP interaction study. Then now the short quick overflow of today's hands-on training session. Here first step is we are going to train the model for the prediction activity prediction. Then we are going to test with our new data with screening of the model for activity predictions. And then we are going to filter the compounds according to their active compounds, with labeling one as active and zero as inactive. With active compounds, we are going to further for the admit property predictions and taking the good compounds for the further execution, like safe compound we are going to do further for the docking process. And from that particular docking molecules, we are going to study the protein ligand interaction using PLIP. So these are the quick intro for our workshop. You will have the hands-on training on all these steps after this session. Now, about all these processes, and you gain the knowledge about the theory part, but what about the practically, where you can study about all this practically and hands-on? So our team is design and introduce one course that is advanced diploma in artificial intelligence and computational drug discovery. So it is a diploma course, and it is a 1-year course containing various type of approaches and the knowledge about the AI, ML, and the bioinformatics and the chemoinformatics and computational chemistry related works and all the aspects. So this course launched by our SilicoScientia team. Then about this course, why this course, and what we are going to provide you in this course. So first, it is a 1-year diploma course focusing on the combination of AI based and the ML based with the biology sector of the world. So in this session program, we are going to give the real world experience about the AI, artificial intelligence, and ML, how make an impact in the our research area, and also in currently various type of research companies and the pharma companies required this AI ML based techniques for the rapid effective work and the less time required. So this is very good program for you guys train here perfectly in this particular area. So it is designed for filling the growing demand for AI skilled professional in the drug discovery. So there is currently in market, pharma companies and the AI related companies are available. So between these two companies, there is a gap between their working cultures and all. And so pharma companies generally uses the more critical workflow for designing of the drug molecules and compound. But AI is a very handful and tricky workflow utilized by the AI companies. So between these two companies gap, we can need to fulfill for merging and for the execution of the quick and smart drug discovery process. So by this program, you guys have to knowledge about how the AI will impact in the both sector of this our companies and both pharma and R&D department and the AML based companies. Then in this course, we are going to introduce you the theory session along with the practical hands-on training with all the softwares and tools and all the curriculum with good technology and the good tools. And it is a very good opportunity for all of you to gain a knowledge and career build in this particular AI and the drug discovery process field. So you can have the so many opportunities like computational chemists, biologists, and the data analysts and the AI scientists and all. So that is a feature we are going to provide in this course. Then about the offering, so this course is jointly offered by industry partner and the academics partner. So this is the beauty of our course, where we are going to patch up the academic partner along with the industry partner. So one who can enroll this course have you will experience the knowledge about how industry works and how the culture and the various type of work happen in the industry along with the academic cultural experience. So it is a very good chance to enroll here. So first is the industry partner. So we are the industry partner as SilicoScientia, and the academic partner is a School of Pharmacy and Research Pune, Dr. D.Y. Patil Nyan Prasad University, Pune. So from our side as an industry partner, we are going to give the all the AI-driven platforms which is used in the healthcare unit, and all the modules we are going to teach hands-on, give the hands-on training, then all the real-world experience we can give in the course. We are also offers access to expert-led training and real-world data set, how to handle the bigger data sets and using this all the ML techniques and all. And we are also the various type of teams like bioinformatics, then ML-based drug discovery, and the computational chemistry work. So all the this aspect we are going to provide in the this course. And as an academic partner, this D.Y. Patil University provides the premier institution it is a very prestigious university as a pharmaceutical sciences in the Pune. So it offers the overall ready curriculum for this course, and also it is very good if you are going to take a diploma for any prestigious university and all. So for the further recognition, you require a very good knowledge and the university for it. So it is very we are going to give the very good opportunity for the enroll for this course. So now the role of AI ML in the healthcare. So as you see, various type of the now the in the various type of market, the AI is booming in the all around in the world while. So first the AI ML is used in the healthcare sector as in the various stages of the drug discovery for the prediction of the drug molecules to particular target, then for the prediction of its adverse drug reaction for the particular molecules with particular target, then it's various type of screening like SAR point of view screening and toxicity screening. So this AI is utilized in the various stages of drug discovery. Then also it enables to high throughput screening of the biological data and target identification. So there is so many vast number of data available on particular disease or target. So how we can narrow down on how we can study as a quick whole large number of data. So we required ML-based approaches for it, ML-based model using various techniques. So it is easy to handle and execute. So it is so that particular analysis this AML works. Then as per the drug discovery process, as I already mentioned, it reduces the cost and time associated with new drug development processes. Okay, so yeah, some processes take a quick half hour, or some take the 1 day or something. So it is very good as compared with the whole process of the drug discovery process. Okay, then it supports various type of predictive models and therapeutic designs and personalized medicine. So various researchers will experience this and try to research on the particular sector of computational biology. So these are the role of the AIML in the healthcare. Now about the program highlight, what we are going to give in this program. So first is a teaching expert from the industry and the academia. As I said, we are going to give the industry experience, which we have the so many expertise in various aspects of the this field like computational biology, bioinformatics, genomics, proteomics, and then the this language system and all. So we will give the teaching experts from the industry and the academia also. Then you have to exposure to regulatory and the ethical framework. So how the data should be transparency maintaining, then how data will show and the data driven privacy and all. So ethical framework and the regulatory aspect, like how the regulatory body works and all. So we are going to teach in this program. Then practical training plus theory session on the Python, Java, and the ML and the bioinformatics tool. You are aware about the Python is a very popular language for the coding and various type of ML-based application. So we will give the practical training for Python, Java, and ML for bioinformatics tool. Then we will provide hands-on experience training on the industry-based softwares and the data sets. So you will get the experience about the lot of bigger data sets and handling and the different types of software which is works on the various type of machine learning methods and the CNN model and the graph neural network. And also we can arrange the guest lecture by industry and academia leaders that will help to gain you more knowledge about this particular sector. So overall, from this course, after the completion of course, you guys will have much knowledge and holding a key command on this AI ML in the healthcare and drug discovery. So next, about program structure. So we designed total 10 modules for our course, starting from the Python programming and application to the last 10 modules. So first is the Python program and application. So you all know about the Python is a language we use for coding. So we train you as a starting basic concept from fundamental properties to the advanced technique. And also in the second module, we are going to introduce and learn about the Java scripting and Java language. Then third module is a biostatistics. So in this biostatistics, we are going to teach you about how we can handle bigger data with the statistically, then its various type of models and algorithms. As a mathematically, we can teach you about biostatistics and biological data sets. Then genomics, proteomics, and structural bioinformatics. So here in this module, you are going to learn about all the experience related to molecular biology, all the mechanism of action, target, receptor, proteins, genes, DNA, and the proteins, all the bioinformatics related things also we are going to give here. Then in the fifth module, artificial intelligence in healthcare and medicine. So here we are going to show you about how artificial intelligence works in the healthcare system, generally like in the pharmacy sector, in the hospital pharmacy, clinical trials, and various type of the data sets. So we can show here. Also, we can show and we can guide about the clinical trials contain so many types of the calculations and the models and all. So using these methods of AI, we can execute this in the healthcare system. And we can going to teach about the machine learning and deep learning in the sixth module. So deep learning is a model where we can study the various type of the model as a CCN model, GCN model, and all. Then data analysis in drug discovery. Then eighth model is the advanced molecular modeling technique in the drug development. It contains so many types of modeling techniques, pharmacophore type modeling, then fragment-based drug design also available in the molecular modeling. Then ninth one is the integrating AI in the clinical workflow. So in the clinical workflow, we are going to teach you about again the clinical aspect of all the AI/ML driven the aspect. Then lastly, ethics and regulatory aspects of AI in the pharmaceutical. So in the pharmaceutical, you all know aware about there is so many type of regulatory bodies like FDA, US FDA, then EMA. So how this body work? What are the ethics are available for pharmaceuticals, and what are the roles of the ethics in the AI/ML base and the merging of both? So these are all we are going to introduce you in this module. So overall, this is a 1 year course, and we are ensured that we will provide you the in-depth knowledge about all these 10 modules. So these are the program schedule structures of our course. Then lastly, infrastructure and facilities. For any course, we require a good atmosphere and environment for the execution and study and the further research purpose. So we are providing high performance computing system, computer power, which is you can easily get work on these computational steps and methods and all. And we are access to advanced drug delivery software. You know about all the various type of software available for a different type of the study, like for chemoinformatics software, bioinformatics software, then AML based software. So these are all software we are going to access you in this course. And also we are providing modern and well configured computer lab for it. For you without any restriction, you can explore and learn here. Then individual workstation for each student for the further study. So this is the infrastructure info. Now the quick program overview about this. So it is a course designed for the 1 year, divided into 2 semesters. Then the mode of this course is we are providing as a full-time regular course. So anyone who is very interested and passionate to growing in this sector of AI, so we are providing the full-time course for all guys. Then instructions means we are going to take this program as a online also and the on-site also. So if you are able physically in the Pune, so we are going to do the on-site course. And if you are not, so we are going to online program. But during the exams and internal assessment, you have to come to on-site. Then about the medium, it is English itself. So this is a just overview. Now the eligibility criteria for the admissions in this our course. So eligibility is any of the graduate can apply or enroll this course, and any life science graduation required for this course. So here is shortlisted and mention the criteria and the eligibility criteria for our course. And the selection method, one by one personal interview we will conduct and select the persons for this course. And for the about the program fee for the Indian student in 1 lakh per annual, and for the international student as per the university policy. So then about the certificate and evaluation. So we are going to conduct the internal assessment plus terms in examinations for these courses. So we will evaluate the performance of candidate based on the grade based performance, like O to F criteria. And after the completion of this course, we are going to provide the diploma as awarded by the Dr. D.Y. Patil, Nan Prasad University. So this is about the certificate and evaluation. Then lastly, career opportunities in this particular sector. So after completion of this course, you will all the experience and expertise and knowledge in this drug discovery process plus AIML based approaches and all the execution. So you have the very ensure about this all role for the performances and all. So there are so many types of roles are available in the pharma companies and along with the IT sector companies. Computational chemist, then the bioinformatics analyst, then machine learning engineer, and the AI scientist. So these are all the various crucial part of the particular AIML based companies. So you can also make a career in the computational chemist. So in the computational chemist, you have the gain and show your talent in the computational simulation models and also the new drug molecule synthesis and the innovation of the molecules. Then also in the bioinformatics analyst, you have to work with more the biological data and the gene sequence analysis and all. So you have the good opportunity to collaborate and to employed as a bioinformatics analyst in the company. Then the machine learning engineer build a model for the AIML based model, predict the drug activities and all. And lastly, the most booming sector, AI scientist. So these are the golden opportunity after the completion of this course as a career opportunity. Okay, so these are all about our course. We launched this course. So thank you so much for your kind attentions and listening. So, if you are willing to admissions and taking the depth in this AI/ML work, please enroll this our course, which is based on AI/ML in the drug discovery. And feel free to contact on our this email ID and the numbers which is provided on the slide. And if you have any questions and query, please ask, and you can also text or message to our team. So thank you so much.
Yeah, thank you, Vikram, for this insightful session about the ML and virtual screening, and thank you for the advanced diploma course information. So, all of you, if you have any questions, please unmute yourself and ask, or just message in the chat.
Hello,
Yes.
am I audible?
Yes,
Yeah,
yes,
Bikram,
you are.
very nice presentation. Actually, thank you for the informative session and all. Actually, I have a couple of questions. Means, means you have generated or designed the ML-based screening thing. But what you explained is that is like a receptor-based drug designing method. So, do you have any means? I'm just asking it. Do you have any means ligand-based approaches means in that ML?
Yes, yes, we can do the generation of models and for the ML base. So you require ligand molecules features like its structural properties and structural features. And some drug molecules have the component of that particular activity, like star point of view study, structural activity relationship. So from that information and from that data, we can also perform the ML-based approaches in the ligand-based modeling. So yes, we can do with ligand molecules.
Yeah, but okay. So that will be one different machine learning we have to train, I think. What is that okay?
Yes, yes, it requires sufficient data for it. So we have to collect
Exactly.
data regarding it. Yes, and after checking data and all, so yeah, if it is good in well, so we can train the model and build. We can try actually ligand based also. Yes.
Okay, okay. And one more thing, in this model, this means protein stabilization things is there, or it is not there. Means Ramachandran plot and protein stability report, those things.
Yeah, yeah.
Yes, yes, yes.
So, yeah, for
Hello,
the,
sir. I'm
yeah,
Pratyek.
yeah. So this Vikram said that, yeah. So for this potential stability and all these things, there is a prediction followed by the validations are there. One that is you are talked about Ramachandra plot, shapes, what check, pro check. Those will be there. Along with that, also we're having the molecular dynamics simulation package. So you can run the short simulation to check the stability of reporting. That is it.
So, sir, in this ML, we are going to initiate from the docking and means MD is also included or it is not included?
You're talking about today's session or you're talking about the course?
No, sir. Today's session means this
No.
ML was.
It is not possible one day
Yes,
to
sir.
cover
Yes, sir.
all this area. So last time,
No, I was I thought that might be some 5 nanosecond or like I something some small
But,
small
yeah,
that.
they will come. So last time we have the first our workshop. We covered how can you use the machine learning in molecular docking. So that we have covered. Today we will go for the machine learning based screening. So gradually, maybe the next new package will come. Yeah, definitely the molecular dynamics simulation will come because in a single day, all the things it is not possible to cover up. Okay. So one by one, maybe the coming workshop we will cover the molecular dynamics simulation.
Okay, sir. Okay, sir. Okay, sir. Thank you, sir. Thank you, Vikram.
Yeah. So actually, I'm getting three questions. Krishna, you are asking about regarding the fee. Yeah, definitely there is flexibility is there. So you may talk to me. maybe text me or email me. Flexibility is there. So for prepayment, you'll get the flexibility. We need to discuss.
Thank you, sir. I will discuss with you.
Yes, yes, sure, sure. And for online mode, you are talking, Maitri, you are talking about today's session, or you're talking about the course, the diploma? You may unmute yourself and we talk about the course. Okay, yeah, definitely. This today is about the course. Yes, because all the calculation, whatever the advanced diploma, that is, you will run. You will be trained in the cloud-based system. Okay, so nothing required from your side. You need a simple laptop. You will be connected to the cloud, so you have to work there only. So there is no infrastructure you need to manage. We'll take care about that. You will give the access. Your all calculations, the model development, execution, everything is done in the cloud. Okay, it's clear to you, Maitri? Okay. And yes, this course, yeah, definitely you will get assistant. 100% placement assistant will be given. That is from the beginning. That is will be trained. That is how to prepare for the interview, how to apply, and the guidance. That is what are the companies are there who are looking for such kind of skills. So definitely the 100% assistance will be there. You will be prepared for the interview, for the placement. Okay, hope you got the answer. Anybody else? Anything? Any question? So if you have any question regarding the course during the session, you can ask, or you can also ask via email or phone number. I'm just typing the email ID here, the chat box. We'll and
So there is one question for free payment also. Have you?
which one? Who else?
There is one question for regarding the fees. We have to pay at a time, or it's there any
Yeah,
flexibility?
I answered already. Yes, the flexibility is there. So just talk to us. We'll give you the flexibility. Definitely. So I have given the email ID and the phone number. So if you have any other questions, you are directly contact to me. During the session, you also ask. Okay. Yeah. If anyone any question.
There is one more question from Priyanka. The session will be online or recorded sessions?
Okay. The course will be both the on-site and online. Those people can come. That is Vikram already mentioned. That is, if you are coming to Pune, your on-site you can access. If you are unable to come, you can join online the classes, and definitely all the classes will be recorded. And the recording, because in some part of time maybe some people miss the class, so they will provide the recording to all of you. So all flexibilities are there. Apart from that, I can assure you that is, if you have any issues, any doubts, so beyond the office hours or beyond the class time, you are always welcome to discuss with us. We are ready to help you in the out of office hours or the weekend. We are ready to help. So that won't be the problem, because as Vikram mentioned, this is a unique course that is involved with the industry, with the academics. So you will be trained, you will be given the environment of the industry during your course. Okay. Okay, Priyanka, you can talk if you want to know more about it. Just call me. We can discuss further. Okay. Anybody else? Anything else?
Dilnawaz, do we have any questions from YouTube live?
Dilnawaz?
Okay.
Okay, thank you so much. Just Omkar, please carry on.
Okay. Thank you all. So now we will move for our third session, which is the hands-on training. Now you have already received the meeting link on your WhatsApp group. You have to join that link. Once you have to attend all whole session of the hands-on training. Once it get completed, you have to rejoin this link to you will get the feedback form that you have to fill. If you are unable to fill the feedback form, you will not receive any certificate. So feedback form you have to fill, and it is compulsory. So thank you, and now you can join your.
Yeah. Om correctly said that is, yeah, Anshu. You will get the certificate. We'll check the attendance of the whole session. If you are attending the whole session and you are staying till end, and at the end we will provide the feedback form. Okay, so definitely you will get the certificate in a week or I mean 10 days. We will send you your registered. Like. Uh, okay, Vivek Patel, could you please tell me which group you are in your WhatsApp? Group number 1, 2, 3. Workshop to group 3. Okay. Manishka just send resend this the group 3. I think if you see the group WhatsApp group 3, you will get the link. Yeah, Aryan will provide the certificate on your registered email. Uh, maybe in the coming 1 week or 10 days. Okay, we'll get okay. So I think in all three groups the links are shared independently. Yeah, so just um, join in your. Okay, anybody else? Somebody raised the hand. Anybody? Question? Yes, Ankshita.
Good morning, sir. Sir, my question is that is any internship opportunity at your institute? Like,
No, unfortunately, there is no internship because this course is always a discounted price, and so there is no internship opportunity at this moment available.
okay,
Because if you compare this course to the any other, because as per my knowledge, there is no such course in India which is a diploma in computational drug discovery using AI. And the second thing is there is no course running with the collaboration of the industry and academic. So this is a unique course, and if you look, some other universities in the abroad, they're providing, but the fees are extremely high. So these fees are already discounted fee. So unfortunately, there is no internship at this moment, but you have the option to pay the fees in few installments. That actually
okay.
is nice.
Sir, any future collaboration with our institute?
Which institute you are from?
Sir, I am from Naipur, Hyderabad.
Definitely, we'll talk. Just you keep in touch. You send email or you can call me. We'll talk. So your faculty there or your student? Uh, Ankshita, your faculty there or your student there? In Hyderabad.
Yes, sir, I am a student there. I am
And
tech
which?
biotechnology second year.
Okay, that's nice. So definitely collaboration might be there. Or during your course also you can during your regular course, biotechnology course, you can attend this course because this is diploma course and there is a option to get online. So also you can opt because as per the UGC norms, you can attend that is with the regular course you can attend for a diploma or online course.
Okay, sir. Thank you so much, sir.
Okay, thank you. Okay. Anyone else? Any doubt? Okay, so we end up this session here, and we'll go to our the fixed group-wise session. We'll start a hands-on session there, and after the hands-on, when we'll come back here, Omkar again.
Once the hands-on is completed from all the groups, and then we will rejoin this link.
Yeah. So that is the thing that Omkar said. We'll go in a 3 groups. We'll complete the hands-on training, and after the hands-on training, we again rejoin together for the final session. In that session, you will get the feedback form. Those people will come back to the session and fill the feedback form. Then only you will get the certificate. After this session, after today's workshop completion, you won't get the link for link of this feedback, and you won't get the certificate. Okay. Okay. Anshu Mishra. Okay. You check your group. If you are in the WhatsApp group, you check it. Okay. Manishka just resend in all 3 groups. Okay.
Sir, I did it in group 1.
Group 1, group 2,
He's
group
also
3
done.
again. Yeah. So yeah. Zulfikar, if you are not getting the link, if you are in a group 1, then you will definitely get it. Okay. So can you share an email? Do you have the email, Manishka? That is, you have the group wise email IDs?
Yes, sir.
Yeah, quickly send to group wise. Okay. Yes, I give some time. She will send you the email through. Okay. Group wise and also WhatsApp email. Okay. Zulfikar is not added any groups. I'll add him. Don't worry. I am adding him in group 1. Okay. Okay. Any issue to related to this? Okay. Okay. The people who are in a different group from our SilicoScientia, please go and
Join this.
okay. Okay. So we are ending up this meeting. Please join your group wise. Okay. So Omkar, you just stay sometime until unless everybody leaving here and joining their respective group. So whoever are from the group 1, group 2, group 3 from our team, please join your respective group and allow them people. Okay.
Yes, sir. You may leave now.
Just wait 2-3 minutes for everyone to join, and then we'll start.
Yeah, Mahima, Parth, just share the Google Colab link to them.
Yes, sir, I have shared the GitHub link. I will share the again.
Share it again, Parth.
Hmm. So, Mahima, let's start. Or should we wait one more minute?
Oh, let's wait for a minute and then we'll start.
Okay. Yes.
But we'll start.
Let's go.
Yeah.
So I will be sharing my screen. Mm, Is my screen visible?
Mm, Not yet. Yeah.
Yes. So I'm sharing the link again of the GitHub for all the new joinees. So just please click on that link and open that GitHub. So this page will open. So here you will find about this GitHub, the README file, what data this GitHub contains, and you will also see the link, that is Collab Notebook link. You all have to click on this Collab Notebook link. There, after clicking on that, a notebook will appear. So in this notebook, all the codes, all the dependencies which we need to for today's hands-on session is present there. So we will be doing all the steps one by one. So first, if you have not used the Colab, just sign in with your Google account and then just click on connect.
Is everybody following? Are you able to access the link, everyone?
Yes, you are able to access the link, yes?
Yeah.
Yes, ma'am.
Okay. Are you at this Colab notebook?
Anshu Mishra is saying no. Yeah, Anshu. Can you please tell me what issue you're facing? You can unmute or maybe you can just. Okay, no network. Okay. Try switching your network and let us know.
Okay.
Because I guess everyone else is able to access. Just try switching your network. Okay. Uh, so should we start part?
Yes, we should start. So,
So in our previous session, like the theoretical sessions, you have already given, you have been aware of your what work this workshop will bring you for. So in this hands-on training session, we are going to mainly deal with the high throughput virtual screening with machine learning. So already Vikram in his section has explained thoroughly what is virtual screening and what are the steps we are going to follow in this hands-on session. In a very brief, I will try to make you understand what is the workflow, each steps we are going to follow in this process or in this notebook. What are the steps we have? So, can you go up? Yeah. So first will be setting up the environments and installation of all the necessary dependencies. So many people have already previously in our previous workshop also have attended. So there were many dependencies or whenever you're using this collab notebook or some ML based things which are having certain uh, codes and everything. So you need to set up the environment for that. And there are different packages for different tools which we need to install. So first step will be totally upon setting up the environment for our for the workshop experiment, and then all the necessary dependencies packages which we have to load. That will be the very first step. Then it will be, we will train our model, machine learning model to predict the compound activity. So this training process will be done from the known data. Okay, we all the necessary data sets, CSV files, data, everything is provided in this same collab notebook itself. So second step will be the training the model. Third step, then we will, using the training data, we will screen our chemical library to find the potential hits. Fourth step will be predicting the test data, the test compounds, which are not a part of our training data. We are going to take totally new molecules from different data set, different database, and we are going to predict the activity uh, of this potential candidates, and then based on the ADMET properties, that is absorption, distribution, metabolism, excretion and toxicity properties, we are going to filter the compounds from the whatever potential active compounds we will get, that we will filter. Then we will prepare the final candidates for the molecular docking. Molecular docking will be performed by Autodoc Wiener tools. Okay, Autodoc Wiener. And then we are going to prepare our protein. We are going to prepare our ligands, and we are going to perform a automated molecular docking using Autodoc Wiener in this collab. Then we are going to analyze the docking results, simulation docking results, and predict the binding affinity. And also we are going to find out the best pose from different poses it will generate. And the very last step will be to find out or to see what are the protein and ligand interactions of our best top candidate molecules. So in all, there are 7 steps in this collab notebook for this hands-on workshop. One by one, slowly and steadily, we are going to go through each and every step, and hope you all will follow us. And throughout this session or throughout this step, anytime you have any doubt, you are stuck somewhere, you're unable to understand, you have any questions, you can just unmute yourself, you can raise your hand, otherwise you can type in the chat box where you are stuck. We are there to help you. So I request Part to start the session, the hands-on session. One by one, he will run each step. He will. We are gradually also going to explain what is happening in each step. So yeah, Part, let's start.
so let's begin. Firstly, we will set up the environment and the dependencies. So dependencies and setting the environment is the main crucial step for us before we start any model training and anything. So I can show you the code also. So in this, the main packages we have, which we are going to use are RDKit
Parth
for
and
the,
Renu.
yes.
So just to do one thing, go slowly and ask them to follow yourself.
Yes.
because this everyone, actually our objective to everyone should do their own. If anyone has any issue, you are always asked to stop your interface and share your screen if you have any issue and solve it. Okay, okay, go ahead.
Yes, sir. Yeah.
So majorly we have the four major dependencies which we are going to install using the pip. Pip is a Python installer. These all are the Python packages which we are going to use. The first is RDKit. RDKit is a computer chemistry package. This is used for reading the multiple file formats, converting multiple file format into different file formats. For now, what we are going to use using RDKit is generating the Morgan fingerprints. So using RDKit, we are going to do that. After that, at the admit AI, which is used for the pharmacokinetic property prediction. This is also a Python package. This is a built-in model. So we going to use that model and predict the pharmacokinetic properties of the molecules. Then OpenBabel. This is also a strong package which we are going to use. This is used for the file conversion. So basically, we are going to use the smiles for the training of the molecules. But eventually, for the docking, we are going to convert those smiles into a 3D structure, PDBQT format. For that, we are going to use the OpenBabel package. And the last is the matplotlib. You can understand by the package name also plot. So we are going to plot some plots using that package. So in the first, you have to just click on this play button and click on this. It will start first cloning the GitHub. After the cloning of the GitHub, it will start the installing of the packages. So it will take couple of minutes. If you have any doubts regarding the package or anything, you can ask.
Yes, you have to just click on the cell where it shows running. You will see a button, pause like button. Just click there. It will start installing all the dependencies, the very first cell. So is everybody following?
Everyone is executing this step now. Before that, you have to connect your collab notebook. Everyone has done that also. After that, you have to
Yeah.
click on this play button.
So part
Is there
someone is
any?
saying to repeat it again. So can you please repeat this step again? Explanation and this thing.
Okay. So in this first step, first we are going to clone the GitHub which from where you have opened the collab notebook. All the data is present there. So we are going to clone. So for that, we are using git clone command and the GitHub link. Clone the GitHub. Second, we are going to install the dependencies using the pip. Pip is a Python installer which we are going to use for installing the different Python packages. The major packages which we are going to use is RDKit, AdmitAI, OpenBabel, and Matplotlib. RDKit for the fingerprint generation, AdmitAI for pharmacokinetic property generation, OpenBabel for file conversion, and the Matplotlib is for plotting the different graphs. And after that, if you have understand this, just click on this cell. It will start executing. You can see there also executing for 2 minutes. It will take couple of minutes. Then we are ready for the next step. Our environment is completely set up. We have installed all the dependencies which we need for this particular hands-on session. So yes. This is completed. There will be a pop-up to restart the session. After everyone will get this pop-up when all the dependencies are installed. Just click on the restart session button. It will restart your session with all the dependencies that are installed. So session is installed. Everyone has completed. We will wait.
Yeah, everyone is on the same page.
Yes. Just gives your thumbs up. then we will start move to the next step. That is very crucial step for now, which we are going to train our model.
Yeah, it will take maybe a few seconds to restart the session. So
Yes,
just wait,
only
don't do anything, it will restart.
yes.
Okay. So yeah, we are waiting, Krishna, we are waiting. So next step will be the training of our model. So we have used
So
this training data set from the binding DB database. We have taken approximately, I guess, 3,000 molecules for training the data. And they are all based on
natural
a specific
conformity.
target, that is tyrosine kinase. Okay. So their smiles we have taken also for training the data. The activity is very important. Any kind of activity will work, like IC50, PIC50, KI value. So here we have taken IC50 as our activity. So for training the means how much your training data is bigger, large in number, that more will be your model will be trained in a very good way. Okay, why? Because it will get lots of features to learn. Okay, for the learning of the model, the the large data is helpful, and the more it is bigger, the more features it can learn. The model can learn from the features, and more in a very approx or good way, it will test the data or predict the activity. For that purpose, whenever any model, any machine learning model you want to train, whether this target specific or any kind of model, the very basic thing will be to search for appropriate data with proper features, with proper labels, with first of all, you have to design or you have to think of what kind of model you are developing and what features you will need. Okay, so based on that, you will choose your data. So large number of data is always like good or suggested to take large data for training. So yeah, everybody has restarted the session. Can we go ahead for training? So I'm so sorry, I'm unable to run. Still stuck in the same page. Can you check your this thing? Check your internet, please, and then again connect to the same page. What issue you have? Can you unmute and ask? Krishna?
Ma'am, it is showing you must restart the runtime in order to use newly installed versions.
Can you share your screen?
Yeah, ma'am. 1 minute.
Pat, can you stop your screen sharing?
No, it will share.
Yes, Krishna, you can share your screen so we can all hear you.
Yeah, ma'am. 1 minute. Screen sharing option, I'm not unable to see here the screen sharing option.
Okay.
I stop my presenting.
To allow him.
Yeah. Yeah, it's
No? Okay.
yeah. It is showing like this.
Just you have done after you have done the restart session, this will come. So
Okay.
you have done the restarting. So you have click on the restart session, right?
Yes, I clicked.
Then it's okay.
Yeah.
It
Okay.
come before the restart.
Yeah. Okay.
So we are ready to
Fine.
move to the next, I think.
Yes, everybody has completed the first step.
Okay, more thumbs ups, like yes.
So, I don't know whether I'm pronouncing it correctly. Malari VC, you are stuck, or you want to share your screen, or you want to unmute yourself and ask where you are stuck. Okay, he needs a short recap of what has happened.
Till now,
Part
just we have,
can. Mhm.
yes, my screen is visible, right?
Yes, yes, yeah.
So you are at this page, right? What's your name? I forgot.
You can call him Malar,
Malar,
I guess.
Malar Vizi. Yes, okay, yes, you are at this page. So you just have to click on this play button. You see this play button? Uh, then you won't able to see the code. Click on the show code, then you will able to see this code. I think because of that you have not able to grab that. So if you click on the show code, you will see this code. This is the first play button. And after that, you just click on this play button, run cell button. After this, it will execute for 2-3 minutes, and then there will be a pop-up to restart your session. Just click on restart session. You are ready with the environmental setups and installing all the dependencies which we need for the today's session. That's it. That we have done in the first cell, first step.
So he has followed. So yeah,
Yes.
we are moving to the next step, that is training the model. Yeah.
So as Mahima has already told to you, so we are going to train the model in this step. So in this step, we have taken 3600 compounds which are labeled. So mentioned there, the data which we have taken is labeled, and they are labeled as active or inactive. So I must show you the data, so it will be easy for you to understand. Second, in the workshop screening, you can see the screening training dataset. So in this, wait, see, so this is our data. You see this column label. This one represent that this compound is active. And if you move forward in this data set, somewhere around just see this zero means this compound, or you can say this smile is inactive. So we are providing the model, the label data set. That is the data is divided into, we have taken 1800 compounds as active compounds and 1800 compound as inactive compounds. And they are labeled as 0 and 1. 0 for inactive, 1 for active. So total
Okay,
3
so
and
this
3000.
active, yeah, this active and inactive is totally depend upon the biological activity which we have taken from the binding DB dataset. There we will get the biological activity, and based on the biological activity, the range of the highest and the lowest IC50 values. So the less the IC50 value, the more the active is the compound. So that is the phenomena. So that concept we have applied, that the IC50, the compounds with very less IC50 has labeled as active compounds, and the compounds which are having higher and more IC50 values have labeled as inactive. So for active it is 1, and for inactive is 0. So that way we have divided the train set into active and inactive molecules. Yeah, Parth.
Yes, yes. So that's how we have labeled the data. So after labeling, what we are going to do? What we are going to do in this model training step is we are going to generate the fingerprints for the smiles. Why? You should ask why we are generating the fingerprints. So we are humans. We can understand why what this smiles giving us, which properties we can understand by that, but the machine can't. So for machine, we are going to convert this smiles in a numerical binary format using the RDKit, and we are going to convert into a machine readable format, that is binary codes these smiles, so machine can understand and predict that these features are important, that makes this smiles particular smiles inactive or active. So this is very important step. So we are you using RDKit for generating the organ fingerprints. Then after that, the third step in the training, that is splitting. So we are going to split our training data, model data into training set and the test set. So in this, we are splitting in 80-20 ratio. 18 is our training data and 20 is our test data. So out of 3600 compounds, we are dividing 80% in a training data set and 20% in test data set. So after that, which model which we are using? So there are couple of there are many models which we can use, but based on our understanding and our data, we have used the random forest classifier. So why the random forest classifier? This is a tree-based machine learning model in which we make the decisions. In single tree-based model, there is only one decision that is made. But in a random forest, it's like a community. So there will be a multiple steps, multiple iterations in which we make the call that this by this feature this compound is active, this compound is inactive. So based on the voting, the higher the vote for if there are hundreds of votes, if there are hundreds of steps, and if we get 80 votes in positive that this molecule is active, and 20 votes this molecule is inactive, then from the majority vote in is active, so we have considered this molecule as an active. So this is a very good model. So after that, we will test our model on the test data set, and we will see the results, what the accuracy our model gives, how it's performing, and that's all. And after that, we will see the rf model.pkl file. That is our model file. So we don't need to train again and again to use that model. We will get the pkl file, that is model file. We will use that model file to predict the unknown data set, which we are going to in the next step. So you just have to click on this step, this cell. So after clicking on this, this will load the screen data CSV, then generate the Morgan fingerprints, then split the data, load the random forest classifier model, and then it will predict on unseen test data, and then it will give us. So the model training is done in 11 seconds. You can see our model is made, and with an accuracy of 0.94. So it's a very good accuracy by which our model is trained. So you can see in the precision also, the model is able to identify inactive compounds with a precision of 0.95 and active compound with 0.93. So this is also a very good precision, and accuracy is very good, 0.94. And we are get the model file, that is rf dot model underscore model dot pkl file. So everyone is
Okay.
following.
So
You can see
yeah.
you can see at
Uh,
in this left rf model dot pkl file which we have generated. Yes.
so everybody has, I guess, trained the model step have been done. So as you can see, the accuracy score of our model is very good. It's all 0.94. But what will happen, or every time you will not get the good result for the training of the model. You will face many difficulties. Maybe your model will overfit sometime. Maybe your accuracy score is very low. So what to do in that scenarios? Okay, so we have majorly or prominently, it is all depending on the data which we are taking as input, right? So first of all, if your model is suppose giving a very low accuracy score, so you have to optimize the model using your data only. So means you have to, now we have used as 80-20, as 80 active and 20. It gave good results. But might not every time it will give the same result based on different data I'm talking. Okay. So then you have to check, split your data into 70-30, maybe 60-40 sometimes. So this kind of combinations you have to try on your training data, training your model, and then you have to check the parameters, how your model is learning. Okay. So in that scenarios, you have to check your data, you have to optimize some features into your code, and then you have to come up to a certain accuracy where you can, your model is trained in a good way. So these are the parameters, the precision, the recall, the FI score, and all these things you have to check whenever you train any of your models. So yeah, here we talked about Morgan fingerprints. So I don't know, you people are from, like, all are from the same background, chemistry background, cheminformatics or not. So I'll just explain in a very short this how this Morgan fingerprints work. So these are a type of molecular fingerprints which we use mostly in cheminformatics to represent the structure of any molecules. And what they do, they encode the presence and counts of the substructures within a molecule. So what happens here is each molecule, suppose each smile is treated as a molecule, and that the fingerprint generation process, this Morgan fingerprint generation process, it starts by considering each atom of your whole molecule. It consider it as a central atom, and then it starts to based on the radius, the adicate, based on the algorithm, it what happens, it explore the circular neighborhood of this central atom, and then based on the radius, it gives us, it counts the fingerprint, and then convert it into a binary code as a binary, which we call as a fingerprint. So that is how this Morgan fingerprint is calculated. We have calculated here it using the adicate. So every bit will represent the presence or absence of this specific substructure. So how that this is how we have calculated the Morgan fingerprints here. So yeah, Pat, you can continue. I'll just
Any other
ask
doubts?
quickly that everybody has trained the model, or anybody is having any doubt in any step or any code, anything, any questions you have to ask.
You can ask. And if you one more insight I want to give, that if you are a model giving a low accuracy, you should first increase the number of the data you have. Higher the number, higher the diverse data you have, the more you have the chance to build a great model. So please collect a good number of data set which have diverse type of molecules. So a good model can be made with a high quality of data. So quality of data is a very, what we say, prerequisite. Quality of data is the main thing which we have to look before any model development. Uh, so everyone has completed this step. No doubt still here. You
Yes,
all
you
have
can
seen
go
this
forward.
rfmodel.pkl file on your left.
You can
So
download it
this
also. This is the modern.
this is the model file which we are going to use to predict in our next step. So let's move to the next step. Mahima, we should move now.
Yes, yes. Yeah.
Yes. Okay. So the next step is, for what we have done, we have built a model. So we have to use that model also. So we are going to use that model on completely unseen data. This data is not used in the training purpose. This data is never used. So we are going to use that data to predict its activity, that molecule is active or inactive. So we are going to use this model. We are again going, let me show you the data. So this is the data. This only contains the smiles. You can see smiles and names. There is no labeling here. See the whole data. There is no labeling, which was there in the screening, the training data. There was a labeling like 0 and 1 for each smiles, but there is not. That we are going to predict that this molecule is active like 1, this molecule is inactive 0. So we are going to use that model which we have developed now for this data and predict its activity. So what we are going to do for that? First, we are going to load this model. Then we are going to load the data set, the predict data CSV. Then we again going to convert those smiles in the Morgan fingerprint, so machine can understand. It's like a barcode which we are which we see in all the products. That's also understand by the machine now. Like that, Morgan fingerprints are like that, which machine can understand about the this is a small molecule, this properties this molecules have, like that. After that, we are going to predict the activity label for the data set. The label as 1 for active, 0 for inactive. You know already. And then predicted labels.csv. This is the file which we are going to get after this step. So let's click on this run button. Uh, you can see. Should I show you the code also? You can see clf job live.load. This in this step you are going to load your model. This is the step where the input data for which we are going to predict, that is happening. The reading of the CSV file is happening using the Panda. Panda is a library in Python which we are which we are using for reading the CSV file. Then this is the step which are featurizing smiles, like generating the Morgan fingerprints. You can see we are using the first we are camp mall from smiles, this which we are using to read the smiles. This is rdcat package. So the rdcat command by which we are reading the smiles, and by this allcam dot get Morgan fingerprints, by this we are getting the fingerprints of the smiles which we have read using the rdcat. Then all this we are going to predict. This is the prediction step. After the Morgan fingerprint generation, we have just predicted the using our model and save in the predicted label.csv. So this step is also completed in 3 seconds. You can see we have generated our model in 11 seconds. That is pretty fast. And we have predicted the data also. So I should show you this predicted label.csv. You all can see in on your left. So now you can see there is one column that is added, that is label column. For each molecule, for each smile, this column is added, and you can see that this first compound is active compound, the second compound is inactive. Like that, all the molecules have been predicted as active and inactive. So this is the virtual screening. This is the model development. This how fast we have done. We have predicted thousands of compounds just in seconds. So after that, there is a crucial step also after predicting. So everyone has followed this step. Anyone has any doubts or anyone has facing any
Okay.
difficulties in this step?
Quickly, I'll tell you what. First of all, what we have taken in the training data, we took the binding DB data set, and it was totally a targeted database on tyrosine kinase. So the kinase protein database we trained the data. Okay. Now for the predictions. Okay. What we did, we have taken a totally unknown data. Unknown data means which was not into our training data, but it is a taken from the Lotus database. Lotus database is a database with lots of compounds and millions of compounds of all the natural compounds. Okay. So what we have done is we have taken a data, trained the model with known parameters, and from that, using that information, we have screened the compounds and we have predicted the activity of natural compounds towards our target, which are active. As Parth has rightly showed, that some of the molecules predicted as active came as active and some as inactive. So this is where you can use. Okay. So you train the model with another known data, and now you can use any unknown data databases such as the natural compound databases, or you have some very specific databases like entire ChemDL you want to screen. So you want to find out that which compounds are active for your target. So in various projects, in various diseases predictions also you can use. Suppose we are working on some cancer disease. Okay, we have a large database such as ChemDL, which have like millions of compounds. PubChem. You have to take all the data or all the small molecules from 2-3 different databases, and you want to find out that which compounds are active for your target. Okay, suppose ACHE, which is a very known target for Alzheimer's disease. Okay, so you just don't know from this huge chemical space how to come up with molecules for your specific target. So there, this models will help you. Okay, we can use a very suppose maybe thousands of data we can use and train our model, and then we can screen large millions of compounds and find out the active compounds for our particular target. So there is the you can see how this virtual screening through AI has helped us. Okay, that is the power of machine learning and this AI, which has have saved us job into from like what you can say in a very less time we are screening such millions of compounds. And this kind of things we have already done into various projects in our SilicoScientia also, where we have used the millions of library data sets and found out the potential compounds for our target using such machine learning approaches. So this is a very basic model which we are showing today. We have used a very limited data set because of the time and all, but it has a very a huge potential. These models and the predictions, everything has a huge potential to screen out the compounds. So that is what is the potential of using modern machine learning and AI technologies into traditional computational approaches. Yeah, but is everybody on the same page? Can we go on the next step? Where are these data sets available? Okay, so these data sets are available online. Lotus database is freely available. Then binding DB is also a database which is freely available. You can access, and you can also download the compounds into SDF, or you can get the CSV also. There are different parameters where you can advance search and download this database according to your target, which target you want to work on, which protein target you want to work on. You can just simply go into binding DB, and then you can search there into the search bar. You will get the database, then you have to filter out based on which organism or maybe the biological activity which you want to take, and you can just download it from there, from the training for the training for this workshop. Other than that, there are many. There is ChemBL, there is PubChem, there is SelectChem. There are different databases available from where you can take this data. Okay. I guess I have answered the question, so I can go forward.
Should we move forward? So the next step is filtering for the predicted active compounds. So we have predicted the data. We have getting the active compound as well as inactive compound. So what we need further, we are going to narrow down our compounds. We have so we will keep only the active compounds and leave all the inactive compounds which we don't need, right? So first we have predicted. Now we are keeping only active compound. So you can see there is a funnel type thing. You are downing the compound size, the library size. So this is what we are going to do in the till the final step. We are going to narrow down the compound size, and we will get the top molecules which we are going to, which someone will go to the next step, the preclinical like that in the next futures. So in this step, we are going to predict the active compounds only. The predict we have predicted, but we are just filtering out the active compounds. So we will click on this cell, and we will see that 2189 compounds, active compounds are ready. You can see this CSV also made filter compound.csv. So all the active compounds are now left only. All the inactive compounds from our data set is removed, and only the active compounds are left now, which we need to process further for the narrow downing, and then we will moving the next step.
So everybody has got their active compounds, filter compounds CSV. All the active compounds will be saved in this filtered
Filter
compounds of
compound.
CSV. Yeah, so you can see we have got 2189 active compounds. Okay. I could not able to see the files.
You have to click on this
Uh,
folder icon, files icon. Then you will
this
able to
file
see all the
icon you
files.
click. Yeah.
Even though I type on that file, I could not able to access that one. Maybe.
Double click on this, then you will able to access this.
Okay, okay. Thank you. Thank you.
Yes.
Okay. So yeah, I guess the compounds, all the active compounds are filtered and everybody has followed. So next step will go. Any questions
So
anybody has till now? Till
you
now, like one, yeah.
till now we have done the major machine learning part. So you should have some questions, right? The major machine
Yeah.
learning part is done. We have built a model from scratch. We have used that model to prediction. So if you have any questions, you can ask now.
Yeah, please unmute yourself and ask if you want to type it in the chat.
You
Please
can
type
type
it because this was all the major machine learning part, and now we have predicted the compounds. And from here we are going to go into some chemoinformatics approaches. Okay, so for this thing, if anybody has any question, please don't hesitate to ask it. Anything it might be. Any silly questions also might help you have some questions in your mind, or you want to ask anything. Feel free to ask. Is somebody saying something?
Okay. Oh, someone has asked.
What is pandas used for?
Pandas is a Python library. So there are many libraries in Python. So pandas is one of them. It is used for reading the files, data files. We have used the CSV file now. So it is used to read the content of that file. So we have used the command PD PD stand for we have import the pandas library as import pandas as PD. So we are using that short form PD, and then we are writing the command pd.read CSV. So this is the command for reading the CSV file. So for that we have used the pandas library.
Yeah. Okay, I got it.
Yes. So let's move to the next step. That is the pharmacokinetic property prediction. That is ADMIT. ADMIT stands for absorption, distribution, metabolism, excretion, and toxicity. So what we are going to do. We have the filter compound CSV, all the active compounds. We are going to predict the all these properties. All there are 22 properties which we are going to predict in different subcategory like absorption, distribution, and after that, using these properties, we are going to again filter out our data. So what we are going to do? Let's first predict the properties. Then I will tell you what we are going to do with these properties. So just run on this cell. It will start
Yeah.
predicting.
Why anybody can answer like why it is why we need to do this ADMET screening or why we want to calculate this property?
Yes.
Would anybody answer that? Why it is important or why is it necessary to screen, check for the ADMET properties?
Anyone here from the pharmaceutical or background?
That
To check the toxicity, actually, to check the toxicity.
We are checking the toxicity as well. Last is T, that is stand for toxicity.
okay, so why it is important? We are going to suppose this compounds for will go for experimental thing from computational. It will move to the experimental cycle. So before going there, these properties are very important to check. Absorption. If you take any drug, how it is going to absorb? Okay. How it is going to distribute it, whether it is going to act on that particular targeted organs or not, whether the toxicity, somebody mentioned that check the toxicity. So toxicity is also of different types. Okay, suppose if we are working on some specific skin related diseases, so carcinogenicity, then the skin permeability, these parameters are very important to check. If we are working on some neurodegenerative diseases, so the BBB, that is blood brain barrier, that parameter is very important and very important to check and keep the thresholds in mind. Okay, so based on what suppose we are working on some tuberculosis disease where the dosage of the medicine which we take, the course is very long, 6 months, 12 months. Someone has to take the tablets, the medicines, the drugs for such longer duration. So liver toxicity is a very important parameter to keep check when we are designing molecules or candidate compounds for such diseases. So based on what you are working for, what type of diseases you are working for, these absorption, and if somebody, if you are taking the drug inside, so will it be okay that that compound will stay inside our body? No. Throwing out that compound from our body is also very important. So the excretion parameters is also very important. What is the half-life of the drug, how it will react, when it will react. So all these parameters are very crucial. If you have just the compounds and just you will say, no, I have found out the compounds, so that will not work. So there are different ways, or there are different tools currently present in drug discovery which we use to predict this ADMET properties. Very hugely or very famous is the Swiss ADME, which is freely available. Okay, but there are some barriers. Okay, to how much compounds we can give into input. Then here we have used is admit AI. Okay. Then there is admit SAR. These are the different freely available tools which you can use for your ADMET prediction. So predicting this and keeping the thresholds in mind when you design or any lead compounds or find any lead compounds for any specific diseases. So that's why the importance of this ADMET prediction and the properties are very important, and to understand them and then filter or go forward into our drug discovery cycle. Okay, so because a lot of drugs fail into the clinical trials itself. Okay, so if we get an idea here itself, so we can try different things. Okay, so that's why predicting this ADMET properties, calculating this ADMET properties of your candidates is very crucial. Also, to study the results and to filter the results is also a crucial task based on the various thresholds. That is also a very important parameter. Also, there are n numbers of parameters you get once you filter this compound. So, which parameters you have to take, we have to consider, not to consider. That is also a very deciding task. Okay, so that is also important. So, this in this workshop we have given a very small snippet of what all possibilities we can touch. Okay, so that is what this our AI diploma course is made for, to give you a very deep insight about all these steps, procedures, how they work, how to decide, how to calculate these properties. If you have the results, then what to do with that results, how to filter out the compounds. That in detail, this our course, that is the AI diploma course in drug discovery with machine learning AI, that is designed for. So yeah, we have filtered the compounds fast.
No, it's executing.
Okay, so it will take a few minutes to complete. So yeah, everybody is on the same page.
So everyone have run this step now. Everyone is-
Okay. Anybody is having any issue, facing any issues? Could not see the data, could not run, or could not understand anything. Okay.
This step is little bit time consuming, but everyone will have complete. I think in 8 9 minutes, everyone must have completed this step. Anyone has any questions, you can ask in the meanwhile it is running.
Yeah. Anybody wants to ask anything?
From earlier steps also you can ask.
Yeah, earlier steps also you can ask. If somebody is unable to follow us from the very steps, they can also tell us, guide them. Okay. What sort of Python skills are required to execute this model effectively? Yeah, fine. Questions
For
maybe.
executing, you must know the basics only. For building, you must know about different models, about machine learning techniques, how to fine tune your model, how to if there are some outliers, detect that outliers and remove those outliers. These are the some crucial steps you must know while developing any model. For the Python, you must know the basics only. For the machine learning thing, you must know deep about it. Has anyone has completed this step, or for everyone it's running? For everyone it's running, I think.
Everyone, it is on the same page.
For
Sir,
me
for
it
me,
is
it's not
not.
ready. Yeah,
Yes, Anshu, what's the
Anshu
problem?
had some issue. Yeah,
Can you share your screen? Or just type in the chat. What problems are you
what
facing?
issue you are facing? You can type it. Or you can unmute yourself and ask. Yes, Anshu. Can you please tell us what issue you are facing? It's still running, right? But
Yes, it's still running. It will take time. The slide you are having is not showing the
um,
slide you are having is not showing the same. I
I
tapped
tapped the
the
phone.
click icon. So then it must showing you this, right? This step must be showing to
Can you
you.
share your screen?
And
How's it going on? My part is good.
Yes, sir, it's going fine. Anshu is facing some issue. Anshu, can you share your screen? Because if you have tapped in, this is not this working. I guess he is not getting. Can you share your screen?
I am stopping my screen. You present your screen.
How to share? How to share? You will get an arrow, upward arrow onto your screen. On your laptop screen, you will get an upward arrow. Just click there and then select. Yeah.
Oh, you are using mobile.
Oh, so if you are using your phone, so how you are running this collab?
Nice.
Can you zoom it?
What else?
Have you
Application warning.
you have done this step filtering for predicted active compounds?
You can unmute and you can talk also. No issue.
You have done this. Okay. Now move downwards. It's running now. It's running.
It's running. It's running.
So this is
Admit will
you are
take 2 minutes
using
to
seeing the
complete.
code.
It will take 2 minutes. Yeah. Can you go down? Can you scroll? It's running. Okay, it's running.
Sometimes it
It will
depends
take a
upon
few
your
minutes to
internet,
complete.
your system. So since it is running in your on your mobile, so maybe taking some time. Okay.
No, it will take time. For everyone, it's taking time. For me also, it is taking time.
Okay. So anybody, everyone is fine and following both of you.
Yes, sir. Yes.
If anybody is any issue, please uh, just stop them and ask. Okay, because we'll finish. Then our job will finish, but ultimately you should take something from this. So please ask the question if you have an issue or if you are facing any problem, ask them to solve it. We'll try to help as much as possible. Okay, please carry on. Yeah,
Yes, sir.
my man participant.
Yes. I'm at the fourth step of predicting the admit properties. You will see the 4 by 5 there. Let me share my screen once again. Uh, it's hanging for me.
Yes. Still running. Okay.
It's still running. In this is you will
2000
see
minus.
the
We are for admit, right?
yes 2189 compounds, active compounds.
Okay. Okay. This will take, I guess, 10 minutes. One.
Hmm. Minimum. For everyone, it's running right. Yes. Okay.
Yes.
I'll share my screen once it's completed. So let's wait for the completion of it.
Anybody is having any doubt or for anybody is completed?
So it's completed for me. You can see everyone is able to see my screen, right?
Yes.
Okay. So this file we are getting admit result.csv. Uh, Let it open. So it contains all the properties which 22 properties for each smiles or each active smiles. So you can see we are getting the molecular weight, log P, hydrogen bond donors. These are the physicochemical properties. Lipinski values, Lipinski score also that this molecule is, I think it's violations, Lipinski violation. This molecule is violating three Lipinski rules, TPSA, stereocenters, AMES, blood-brain barriers, bioavailability, and other admit properties as well. So you can see these all the properties we have calculated just now. So we are going
Okay, so here we have to filter. Here we have to know that which properties come under absorption, which properties come under toxicity, which properties come under excretion. Okay, so these are the basic one. Molecular weight. Okay, molecular weight. So we consider molecular weight. Mostly in most cases we consider below 500. Okay. Then log P, the partition coefficient. Okay, hydrogen bond acceptors, donors, um, up to 5, up to 10 we consider. So these are the various thresholds which we which are available or which we can decide based on what disease, what molecules we are using. And based on these thresholds, we screen out the compounds, filter out the compounds, which is going to be our next step. Now, carcinogenicity we have to check. We have to check the AMS toxicity. Okay, then there is one parameter that is DILI, which is for liver injury. Liver injury. Yeah, this is the clean tox. This is DILI parameters. DILI for liver. Okay. Then HERG is for skin reactions. Okay. So like this, different. That BBB I talked earlier. That is for blood-brain barrier. So these are the parameters which you have to segregate, which we have to get information about, which falls under absorption, which falls under distribution, which falls under metabolism, excretion, and toxicity. So based on this, we are going to segregate it or filter it based on the default thresholds or the ideal thresholds for this particular parameters. That is the next step which we are going to do. So I would like to ask that for everyone the admit screening is completed or not. There are a few people are still for them still it is running. You can just tell us to wait or to move forward.
completed. For Krishna, it's completed.
Completed,
Ritika, it's
completed.
completed. For others, it's
Okay.
still running or completed. For Nilayana,
Okay.
it's completed.
Somebody not completed can tell us that not completed, so we can wait.
Yes. For everyone, it's completed,
It
I
is
think.
mostly everyone is completed.
Yes. So we should move to the next step, I think. Should we, Mahima?
Yeah. Go forward.
Yes. So now we are going to filter the compound based on the admit properties. We are going to choose the threshold for the properties which we are going to type there, and which properties which we are going to use. So there we have defined some properties. We have defined some default thresholds also. So what we are going to do in this step is first we are going to load the result, that is which we have just got the admit result. Then we are going to define some thresholds which are acceptable for different properties which we have set. Then we are going to click on submit, and just we have again got the narrow down the compound size, library size. After this filtering, also we are going to get some more narrow, small compound size. So let's start by running this cell. So the first property, yeah, we have set the default value of 300. You can play with it, but what I say, due to the time construction restrictions, we should just keep on enter. After entering the default value, it will take for filtering out the result. So for the molecular weight, we are taking less than 300 the molecular weight. For log P, we are taking the less than 5.5, less than 5. Just keep on enter. It will take the default values. If you want to play with the filters, you can also play with that. The hydrogen bond acceptor, the less I we are taking the less than 10. You can keep any value. Based on that, your molecule will filter out. So for now, we are filtering out the 2189 compounds, right? So we are taking the default values for now. Hydrogen bond donors, we are taking less than 5. TPSA, to we are taking less than 90. And the
There is a very
Lipinski,
famous rule in drug discovery, okay, mathematics, you can say, that is the Lipinski's rule. Okay, where this parameters, the hydrogen bond acceptor, it should be less than 10, hydrogen bond donor should be less than 5, molecular weight should be less than 500. So all these parameters are already set in this rule. So based on these rules, we are taking this default parameters. If you might have this question that from where are the default values coming? So some are coming from very well-known rules for the physical-chemical properties, and for the ADMET properties, for different tools, there are different value ranges. Okay, now for here we have used Admit AI. So Admit AI has some different ranges and thresholds values for extreme, high, extreme, low. Based on that, we have decided this. If you go to some other tools with ADME, there in their documentations, the default parameters or the acceptable ranges are given, are provided. So if you change the tool, the this default thresholds will change accordingly. So these thresholds are decided based on the Admit AI tool and the documentation and ranges, acceptable ranges for each parameter. Based on that, we have set this default parameters. Okay, so that we can check from our data, and also we can change. For now, we have just kept the default values, decided the default values, and we are using it here. Yes, Parth.
for Lipinski, the default value is 0.5. For QED, we are taking 0.75, the default value. And the bioavailability, we are taking 0.75 as the default value. HERG, we are taking greater than 0.15 as a default value. AMAS absorption, this is the absorption property, we are taking greater than 0.25. So after all this, all these filtering, we have given the threshold for all these well filters. We get total of it's running, I think. Wait, you will see.
The structures.
You will see the structures as well. So it's completed. So we have got, I think, mm, some 3, 6, 9, 12, 15, 19 compounds we got.
That's 19.
Up different
Yeah,
19
19
compounds.
compounds.
So these all 19 compounds are active. These have passed the admit criteria which we have set, the threshold which I've set. So, so you can see first we have got like 4,000 compound, which we don't know about the activity, right? Then we have predicted the activity. We have got 2,189 compounds after filtration, all the active compounds. After that, you see the narrowing funneling type thing. After that, we got 19 compounds only. We predicted the admit properties of all the active compounds. Then we set the threshold. That's how we are narrowing down the compound library range. So from 4,000, we are just now getting the nine. We are just moving down to the 19 final molecules. So you see this. If you gone to the wet lab, you have to screen out manually all the things. You have to test all the things. So it will take how much time? More than a decade. And you have just completed in 1 hour, I think. If you have a good computer, good system, you will complete this in a few minutes, I think. So this is the power of AI. This is the power of machine learning. So you can see from 4,000 compounds, we are just now left with the 19 compounds. So what we
Yeah,
are
and
going to
all
do
these
is,
natural compounds are natural compounds. All these are natural compounds which we can use in our study. So the 2D structures, you can see the ligand smiles is given, the molecular weight, all the parameters are given there. Also, different functional groups you can see which are attached to the 2D structure. So the viewing of the 2D structure is also important to check the diversity of the molecules, to check the important functionalities. If you want to preserve or if you're searching for any say any important scaffold which is important for the activity. Okay, there are different scaffolds which are responsible for different kind of activities in drug discovery. So here within this notebook only we have given the option to view the 2D structure. No need to go to any other foreign tool to download the molecule and then draw or see or view into another this 2D structure viewer. So here itself we have given the option that whatever is filtered, what is finally come up that you can see here, and at a glance you can filter out or you can do your analysis based on the 2D structures, the scaffolds, the different function groups which are present. In a very you can say in it is like a one pot dish kind of a thing. Very fast and very quick with good results. So that is it. So everybody can see on their screens the 2D structures and the compounds which are filtered. Can
everyone is
everybody
following, right?
visualize? Yes.
Yes, yes. Okay.
Anybody having any issue or is stuck somewhere?
Yes, I'm able to
Okay,
visualize.
yes.
Okay, nice.
Everybody is following. That's good. Yeah.
So now moving to the next step, we are preparing our ligand. These all are the ligands molecule, right? Small molecules, the drugs, the future drugs, we can say. So we are going to prepare this for the docking. So for docking, we are using the Autodoc Vina, you all know. So it accept a particular file format. So right now it's in a smile format, right? We have used the smile format for the model development. So right now till this step, it's in the smile format only. So what we are going to do, we have the 2D smiles. We are going to convert this smiles molecule into 3D molecule in PDBQT format. And with that, with 3D, we are going to prepare this ligand also for the docking. In the preparation step, we are going to add the charges in it. We are going to add a particular pH to that molecule. We are going to add hydrogens to that molecule. So this all comes in a preparation step, preparation of the final ligand for docking. So for this, we are using the open babel. We are using the open babel. Should you should see the code. Yes, see this. You see this step. We are generating the 3D. That first we are converting the smiles to 3D. Then for preparing, we are converting the SDF file. We are converting the smiles into SDF 3D format. Then we are converting that SDF file into PDBQT that is acceptable by the Autodoc Vina. We are adding the hydrogen to it. Dash H means we are adding the hydrogen. We are adding the partial charge to it. That is gesture charge. We are adding. We are giving the pH of 7.2. We are setting that this molecule should have a pH of 7.2. We are adding that also. Then in final, we are keeping all the ligand output.pdbqt file in a ligand directory. Ligand directory will come up here somewhere here, and you can see. So this is we are going to do in this step. We are preparing the ligand for the docking purpose. So we are converting, preparing, and saving this. So all of you just click on this run button. It will start executing. It won't take much time. So you can see this ligand folder has come. So it's completed. You can see now in the ligand folders, all the 19 compounds are divided into single compounds and saved in the PDBQT format. All the 19 compounds. If you want to see the file format also, how the PDBQT file format is this, you can see these are all the atoms. This is like we have added the charge. These are all the XYZ coordinate, the 3D coordinates. This is the charge we have added. This is the name of the molecule lig1, like that. This is the PDBQT file format, which we are going to use for the docking purpose. Yes.
If we
Yes,
have
Savi.
a question that does Google
Wait, let me see. Does Google? Yes, in the backend, it's running on the Linux. That's why we have using the Autodoc Vina. It's running on the Linux. Wait for a second. Hmm, yes. So everyone has prepared the ligand for the docking purpose. Ligand folder containing the ligand 19 ligand molecules. Okay, everyone has done it.
Yeah, yeah. Rinku, Parth, and Manishka, just a minute. So once you finish, so then we need to combine all three groups together again. So we'll share the link again in your group to join, and also uh, the instructors. You're supposed to be there until everyone left and joined to the other group. Okay. So, so go ahead.
Okay. So everyone has completed till this step. Let's move to the next step. So we have a protein molecule, the target molecule. We have copied this from the GitHub. So we are docked our ligand compound in this protein molecule, in this target molecule. So in this step, we are just going to copy this protein file in the outer directory. So after running this, we have you can see there will be a protein directory and the protein.pdbqt is saved there. So this is the target molecule which we are going to use for the docking. We will dock all the 19 compounds at the binding site which we are going to define now. So in the next step, just we are going to download the MPI Vina. So run this step. This is nothing. Just we are downloading the MPI Vina for our use. This won't take much time. This is completed within a second. So everyone has completed till this step. If you have any doubt, you can ask also. Okay, Ritika. Anyone has any doubt also, you can ask. Should we move forward? Give the thumbs up. We will going to dock now. We are going to define the binding site, then we are going to dock. Any doubts? Anyone? Okay, no worries. So now what we are going to do, we are going to define the XYZ coordinate of the protein molecule where the ligand molecule will bind. Is anyone has sent any message? Can we use Schrodinger's license tools instead of Autodoc Vina? If you have that, you can use. Why not? You just have to dock the protein and ligand molecule there. There is nothing there else, nothing different there also. You can use the their docking engine, or you can follow the steps here. So thanks. Okay. So we are going to define the XYZ coordinate and the size. So you can see the x coordinate, y coordinate, z coordinate, and the size of the box and the number of poses for each ligand molecule. This in docking we will get the 10 poses of each ligand molecules. So we will going to run this, and it will save some file, some txt file. It's not there. I think it's somewhere there. It saved the txt file where all these xyz coordinates are there. Okay. So now in the next step, we are going to dock. Dock where? Here. The coordinate which we have defined, we are going to dock the ligand molecules, all the 19 ligand molecules at that active site. This this coordinates is we have known the active site of that protein molecule. So there are some software also freely available which you can use to identify the active site of a protein molecule. We have known the active site of
Okay.
that molecule and the coordinates of that molecule. So we have written here the XYZ coordinate. If you have different target molecule, you should have known the active site of that molecule. So you have to write specific that XYZ coordinate of that active site of that target molecule.
1 minute, Pat. Yeah, which PDB ID we are using? PDB ID 3UE4. That is tyrosine protein kinase. This is the PDB ID which we are using. Yeah, you can go forward.
So now we just click on this cell. This will start one by one docking for each ligand molecule with the protein, with the active site center XYZ and the size of the box and the total number of poses we want to get. So now we have get result for the one molecule. So for the first molecule, for which molecule? Lig1. Okay. So for the first ligand, we have got this result. We have got, you can see, we have got all the 10 poses for each for the ligand molecule, and for each poses we have got the affinity, that is binding energy. How good the ligand molecule has bind with the protein molecule, the target molecule in the binding site. This score gives us that. So high, more the negative value of the binding energy, the good the binding is. So you can see that in each lower, when we are going down the pose, the binding energy is lowering. So the first molecule has the best affinity and the best, we can say this is the best pose which we can take in consideration. So this is the best binding score, binding energy, affinity, whatever you say. So this is the interpretation of the result of the docking. So here 3 molecules, 4 molecules, 5 molecules. We have till now have done docking for the 5 molecules. In the result section, in the left, you can see in the results the 5 molecule results has come. You can double click on it. You can
Everybody
see
is
there.
following, right?
You can see there
Everybody
also
on the document.
7.14.
Yeah. So what is the question? Can we predict the binding site using ML? Is this the blind docking? So first of all, it is not a blind docking. Blind docking means you take the entire protein. There is no active site defined. Okay, this config file or the parameters, the XYZ coordinates which we have given, that doesn't come into blind docking. You, what you do is you make your grid. If you anybody have used Autodoc Vina, in there is one step of grid generation. Grid is nothing but it is a kind of a address kind of a thing to where the small molecule or the ligand has to go and bind. So in blind docking, the entire protein is covered into the grid, and we give the flexibility or the permission to the small molecule to go freely wander here and there wherever it find its active site, or the you can say it's bonding there, it will go and bind, okay, into as per the interaction of the atoms and the protein properties and everything. But in this workshop we have not used blind docking, we have used a targeted docking, or you can say rigid docking, where we have defined the grid box that our XYZ coordinate will be 40 40 40, or the grid box dimension will be this much. So that the parameters, the size of the box and the parameters is defined. Now if you will tell me how we have decided this, so here in this workshop we haven't shown you the traditional Autodoc Vina docking, right? So in that, if you check the traditional Autodoc Vina docking, you can find out the parameters, you can change the grid size, you can increase the size of the box. So what we have done is we have already explored this protein and find out the active site coordinate for this protein, and that coordinates directly we are using here. And the answering the next question, can we predict the binding site using ML? Yeah, there are many tools which are used in the market to predict the active site of the binding tools. Okay, there is this very famous tool as called as CASP, which gives you enter the PDB ID, and you will get the different pockets of the active sites. Okay, you will get the different pockets which with the pocket score and the activity score. So yeah,
Drug score.
yeah, drug score, the drug ability score of that pocket. So yeah, there are different tools available which are ML based, and also ML and physics both combined based tools which we can use to predict the binding site. Another traditional way of predicting the active site is from the literature as well. If you have any, if suppose we take this PDB-ID of tyrosine kinase 3UE4, there the attached paper in the PDB, there also you can find out, okay, the residues which are interacting. Using that residues also you can generate your grid box. Otherwise, if you have the co-crystal ligand attached into your PDB-ID, using that co-crystal ligand, also we can find the interaction of that ligand with the small, with the protein, with the macromolecule, using different tools which we are going to do. The next step is that only the protein ligand interaction. So if you have already attached co-crystal ligand into your protein, what we can do is using different tools such as PLIP interaction tool, or there are different tools such as LickPlot. These tools will give you the interaction of the small molecule with the protein and the residues of the protein you will get. So using that residues information also, you can generate your grid parameter, that which size of the box you have to take, so that all the active residues are inside this box. So the grid is nothing but it is a address to a, you can say, a home, as we have different addresses to our home. Okay, that is the address of the small molecule, that where it has to go and bind. Okay, so that is what is this grid parameters. And yes, of course, there are different various ML based tools available which we can use to predict or find the active set. Yeah, so I hope I have answered the question. We can go forward. Path. Yeah, welcome.
So everyone has completed the docking, I think. For everyone, docking is completed for the 19 molecules. For me, it's completed. You can see the results here. For each molecule, you will get 10 poses results. The file also you will get for each molecule. This file contains 10 different poses, different orientation of that ligand molecule. Mm, by which it attached with the protein target molecule. So you can see different binding energies. For each ligand molecule, the first pose has the best binding interaction, binding energy, best affinity, you can say. The lower the binding energy, the best the binding interaction in with the target molecule. So everyone has completed this step, right? Should we move forward?
Yes, I guess everyone is on the same page.
Yes. So now we are going to organize and analyze our docking result. We are going to get organize the result in our docking folder. So just click on this play button. It's completed. There you will see a folder docking output. You will see there is a subfolders all poses. In this, in each PDBQT, you will see 10 poses for each molecule. Then best poses. So by the binding energy, we have extracted the best pose out of the 10 pose for each ligand molecule. So I have already told you that the lower the binding energy, the more the best we can say the poses, the orientation by which it bind with the active site of the protein molecule. So we can say the first pose is always the best pose in this autodoc vena because it has the lowest binding score. So we can this best pose contain this file contain only a single molecule. Thus just only a single molecule. While this all poses contain that file contains 10 molecules like that. This lick 10 for which I have opened the first pose, the best pose. This file contain all the 10. You can see there is a model 4, model 2, model 3, like that. So these are individual molecule. This is separated. Each molecule is separated by end MDL line.
But can we show one of the file in Pymol with all the poses?
Okay.
Uh,
Mahima and Parth, any question is there that I need to answer?
No,
not
sir, not
yes,
so far.
not yet.
We have answered all the questions.
So if you have any question, I am available to answer. Okay.
Yes, sir. Yes, sure.
Wait, let me open that file.
Yeah.
Let
So,
me
so
save that file.
by visualizing, it will be easy to understand for you that what we are talking about the best pose. The first result now part told it is having all the 9 poses as you we will see in the primal session, and how we are finding the best pose that also we will explain you. Using Pymol, we can visualize it.
Yes.
Just a few minutes, okay?
In few minutes it will open.
So yeah, this is the binding affinities of the molecules. Here you can see for all the 10 poses. So more the negative, more the good the poses or the binding affinity we consider. Pymol is not responding.
It will. It will. Hmm. So let's open the file. Let me first show you the protein molecule, with the target molecule, which we have used. Protein.pdbqt. So this is the molecule.
Yeah, this is the macromolecule, the protein. Okay.
So now we are going to open the ligand file. This file contains all the 10 poses. So yes, now you can see.
Hmm, that's it. Yeah.
Now you can navigate the pose from here. See,
See
this
how
is the
the,
same molecule.
yes, this is the same molecule and in different orientations from pose 1 to pose 10, how the orientation of the molecule, small molecule ligand is changing, you can see. All are at the active site only, but the orientations are changing. So from what we have done, what we were talking about, the best pose is that only from all the 10 poses, which is the pose which is very near to the active site that we are finding. And for that, we have made a code that will check all these parameters, and from all the 10 poses, it will give extract us the best pose, because all the 10 poses are not necessary for us. Now, fortunately, here all the 10 poses are at the active site only, near the vicinity. But sometimes what happens in different scenarios, only two to four poses are here, rest 3 4 poses are somewhere else. So these filtering process is necessary. Checking one by one all the molecules is not possible by visualizing and checking that what is the best pose. Okay, suppose we have 10,000 molecules. How we will visualize all the molecules one by one, and then we will check the best pose. So for that, here the approach, the code we have developed, that is very important and very useful to sort out from the 10 poses or poses that which pose is the best pose, and give us the complex of that pose. That is very crucial and important step, which have made the work very easily, a tedious task very easy. Okay, so yeah, that was the aim, to show you this visualization of the protein and ligand, how we are finding the best pose. Yeah, Parth,
Yes. So
continue.
you have seen the result, right? We have from those 10 poses for each molecule, we have taken the one pose, right? So what we are going to do in the next step is we are taking the best pose and the target molecule, protein molecule, and create the complex. Complex means the protein and ligand in a same file. So what we are going to run this code, we are going to generate the complex. What is happening here? Okay, we have to. I forgot to run this step. We have to download the PyMol.
The same PyMol which we showed you by visualization. So here itself, to save the complex, uh, we have to download this PyMol, and then
Once
it
it
will.
is done, everyone. Once it is done, everyone, please move to the link. Manishka, just share the link to all three groups. Okay. So if it is done, wherever done, you can move. Okay, actually, Mahima, team
Yes,
done, or it is need some time?
just last step.
Okay, okay. Once it's done, then move. We are going there.
Okay.
Yes, yes. Okay, sir.
Manishka, just share the link.
So it will take couple of minutes for downloading the PyMol. We are not downloading the GUI version here. We are
And WhatsApp
using
also is share, Manishka.
Yes, Parth, continue.
so we are using the command line version of the PyMol. We are using the commands of PyMol to generate the complex. So wait, let it be installed. In the meantime, let me show you the code. So what we are going to do first, convert create complex. We are going to convert the PDBQT into SDF. We have the molecule. We have convert the PDBQT into PDB format. The complex which will we need we need in the PDB format. Right now we have the file in the PDBQT format. The target file, protein file, and the ligand file. We both those file are in the PDBQT format. So first we need that file to be into PDB format. So we are converting both the files into PDB format. We are using OBabel, which we have earlier used, for converting the smiles. huh. So I think PyMol is installed. So we now just run this step. It will what was I saying? So we have converted both the files into PDF format. Then we will create the complex using the PyMol commands, the CMD version of the PyMol, and CMD load SDF file and CMD create CMD dot create complex file. So we are saving the both the output PDF file and the out in the output file in the PDF format, both complex file. So you can see all the now it's run. We have created the plib complexes it- in the folder. We have all the 19 complexes in PDF format now. Everyone has completed this step.
Yes.
Yes, okay. So now we are moving to the last step of our hands-on workshop. That is why we have converted those simpler individual file into complex, because we have to find the interaction. We have the protein molecule, we have the ligand molecule. Now we have to find the interaction, how they are interacting, which from which by which bond they are interacting, for which amino acid they are interacting, the protein and the ligand molecule. So we are going to analyze that. For this, we are using the plib tool, protein ligand interaction profiler. So this is the code you can see. Just run this cell. It won't take much time. We will get result for each complex. It successfully first it has installed all the plib dependencies, and it's you can see plib is running for each complex. It's running for everyone. It's running, right? If you have any doubt, you can ask.
You can ask if you are stuck in any step.
Yes, it is completed.
Yeah.
Now you can see. Wait, let me go to the first molecule. This is the ligand 8 molecule. The yellow orange part you are seeing, this is the ligand molecule. And this blue region, this blue small parts, this is a protein molecule. This is not the protein molecule. This is the only the section of that protein molecule from which this ligand molecule is interacting. So you can see this blue line. This is a bond. So this bond is forming between these two ligand and the protein molecule. This
Yeah,
also a
so
bond.
there are different bonds, like the dotted lines hydrophobic, the blue one is hydrogen bonds. If you have pi cations, pi anions, pi stackings, all these bonds are you can it will be shown using this Philip interaction diagram. That is very important for us to know whether our ligand and protein which has been docked as docked in the same place or not, what kind of interactions it are forming, and based on the type of the bonds we can decide the whether it is a strong bond, what is the role, and these all plays importance in the activity also. If you can see, if you take this all further to the MD simulation step, okay, there also these bondings and the interactions are important. There we can get the detail about each interaction, how the protein and ligand are interacting. If the hydrogen bonds are more, the interactions are very strong. Okay, how this all will affect the further step in MD simulation. You can see it all in very detail and in the very atomic level. All these was into the protein level, and docking means it is not that much flexibility. Some restrictions are there. MD simulation, there is no restriction. The protein is free to move, the ligand is free to move. that is all the detailed steps, the next steps. Okay, so for now we have concluded or come to the end where we have uh, find out the interaction between our protein and ligands, which we have docked from the active compounds and admit filtrations. So that is it. Yeah. Fine.
So you can see there is a PLIP report folder has made. This is the cysteine molecule. Let me show you the interaction file. So you can see the hydrophobic interaction, the hydrogen bonds. These two interactions are coming here. You can see the residue number, residue type, residue chain, ligand molecule, ligand type, ligand chain, and the distance also. These are the ligand coordinates, carbon index. This is protein carbon index. This is the ligand coordinates. This is the protein coordinate. So this is the TXT file. So this, if you're not able to understand what it is, you can see here that this residue, that valine is interacting with this ligand UNK and making the hydrophobic interaction. And here hydrogen bonds, that MET 318, the residue number of that, and it is present in the chain A, is making a bond with the UNK, and the distance is that, angle is this, and the coordinates of that interaction is also present. So this is all about the PLIP. This is all about the workshop. So what we have done, we have done, we have downloaded the dependencies. We have then train our model, our own model from scratch. We have developed a good model with 0.94 accuracy. For that, we have taken 3,600 compounds. After that, we have predicted the activity of the unseen data. Then after that, we have filtered the active compounds only. Then for those active compounds, we have predicted the admit properties. After that admit property prediction, we have again filtered out the ligand library based on the thresholds of the properties which we have chosen. We have gotten 19 ligand compounds. Then we have done the docking, protein preparation, ligand preparation. Then we have done the docking. After docking, we have created the complex using the best pose, which has the lower binding energy. Then we have done last step, that is the interaction profiler. So this is all about the workshop, these are all about the steps which we have done in this workshop. If you have any doubts in any step, you can ask or you can reach out to us.
Okay. So I guess everyone has followed all the steps, right? From start to end, everybody has completed. Is everyone completed?
Yes.
Yes. Okay.
Yes. Okay.
Okay, that's really good. Okay. So yeah, that was the hands-on session for using ML, machine learning, and AI in drug discovery for virtual screening, as Parth has rightly mentioned. So this was very basic level model development and prediction, and very few steps we have involved. Using machine learning and using AI in with this traditional drug discovery approaches, we can do wonders in this field. Okay. Only we need is the right knowledge, the right approaches, how to use it, how we can build on it. That important, that knowledge, the basic knowledge of the concepts, basic knowledge of machine learning, how to use that traditional knowledge and apply it into machine learning. That gap we have to bridge, and for this more detailed understanding, definitely we will bring more such workshops. Also, our course with DY Patil, the 1-year AI diploma course, has the potentiality of making you understand the uses of this AI ML in drug discovery, and we have designed the course as to make you, you can say, a very good into this field, where you can develop your own model. You can solve your drug discovery related issues using the power of AI and machine learning. So I would thank you, everyone. I would like to thank for spending your time, your patience to cooperate and follow us throughout the session. So it was a very good experience to uh, host a workshop with you all. So yeah, for future any queries you have for the workshop related or the course related, if you have any questions, you are every time free to mail us on the email ID sir has said. So yeah, quickly I want to wrap up this session, and you are requested to join again on the link which is provided on the WhatsApp group. So there we will conclude, and feedback form will be shared, and you have to fill the feedback form. Those who will fill the feedback form will receive their workshop certificates. So yeah, let's join there.
Let's join there. Okay.
The diploma course. So if you guys are interested, you can ping us to us. Let us know.
Sure, I'll talk about this. One other group is joining here. 15 minutes because they're taking a little bit of time. But in between, we can discuss if you have anything. Also, we are organizing 3 days workshop in Bharati Vidyapeeth, Pune, 29th, 30, 31st July. So if anyone is interested, we go offline and more details. So it is SilicoScientia and Bharati Vidyapeeth. So whoever in Pune or nearby, they can join physically. So you can register there. So definitely fees are there. So yeah, that is actually Manishka. Mahima is there, no?
Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
Okay. So all I can wear, am I right? Okay. So I think people are coming. Just wait 10 minutes.
Yeah, Vivek, you can find the details of the offline workshop on our LinkedIn page also. Definitely we will share here also.
Well, yeah, the offline details which share in WhatsApp group. So you may register there. There is some fees, but it is nominal fees. I think not sure. For 3 days actually. 3 days, so you will get in great exposure there also. Okay, so thank you so much everybody for this joining this workshop. So before conclusion, this session will be concluded by Anirudh. But before that, I would like to just mention few things. One thing is that whatever you have learned today, so half day it is not sufficient for to understand all the. So this is just the idea that is, yes, we can do all this thing. And another thing, here we have given you the code. You are not writing your code by your own. Okay, so because it is not possible to, everyone are not expert in Python programming, and the machine learning means the Python programming you should know, because most of the packages of the machine learning are coming in Python. So that's the reason actually we have launched this course, that is the advanced diploma. So this course will start from the basic thing, So if you opt this course, so you will be expertise to create your own code, develop your own algorithms. Okay, so you can write by yourself the code, your algorithm based on your data. So you'll be trained very well in the Python, and you'll be trained to develop the machine learning models, the Java, definitely the computational drug discovery, the advanced level, like from structure-based to ligand-based, docking, virtual screening, pharmacokinetics, QA, pharmacodynamic simulation. So all these things actually will be covered in this diploma course. So another thing is that if you are doing any other courses, regular courses like final year BPharm or final year graduation in any subject, or in masters in any semester, maybe first semester, first year or second year, or maybe there is PhD scholar, or maybe the faculty, or maybe any professional, you can join this course, because this is a diploma course. So along with the regular course, you can join. And also you have kept the flexibility, that is the on-site or online. So if you are in Pune, you can join there. If you are not able to join, so you can join online. And also from the abroad, few admissions already done. I think around 50% already done. Among them, the few are from abroad also. So if whoever in this session from out of country, out of India, they also can join this course. So that is there. And definitely after the course, the job opportunity will definitely increase, because today's the market is the application of artificial intelligence in any field, and also in the drug discovery. So if you have any query, anything, you are always welcome to discuss with us regarding the course, regarding the fees, regarding the duration, regarding the mode of conduction, etc. Have anything, you just welcome to ask us. Yes, for us, we do provide the protein engineering course. So training will give. You may write, or you may call me. We'll discuss. Okay. Apart from that, I mentioned that is we are going to conduct one 3 days training, the 3 days workshop, the offline in Bharati Vidyapit. And Bharati Vidyapit. So whoever in Pune or nearby Pune who wants to join, so 29, 30, 31st, 3 days. So you can register. We'll post the registration details in the WhatsApp group, so you can run it. Yes, Julfikar, this course will be first you will be trained with the traditional computational drug discovery, and then how can we use the machine learning models to do the same thing. Like in the last session, what we have done, we know the docking, the molecular docking is the order of Vina. That is the traditional software and the conventional way. Now, how can we use machine learning to dock millions of molecules at a very less time? Okay. So that docking model, the machine learning model for the molecular docking will be developed, and it will be implemented to dock the large number of molecules. Similarly, the virtual screening, similarly the pharmacokinetics, similarly molecular dynamic simulation is also true. So yes, it is the traditional software will everybody can learn everything, but how can you use this traditional using the machine learning? How can you develop the machine learning model for these tools or these softwares? That actually, this is the main objective. Okay, so tomorrow, once you complete the course, then you will be expertise to develop your own model for any computational drug discovery, like de novo design, like the screening, like docking, etc. Okay, so it is not simply like that. Is you will learn all these things. Okay, so this course, can I do high impact or contract discovery, especially I would like to incorporate my project? Yeah, definitely. If you go through our website, you will see that in last 2 years, we have already published around 16, 17 research articles in a good impact journals. So that is its initial. Now gradually you are maturing. So whatever the project you can see in our papers, now it is better you will see. So in this course, actually from day one, you will be along with the theoretical, it will be the part of project and the hands-on practical sessions. Okay. So our objective, all the students, all the projects of each student will be published in a good journal. Yeah, so that is our main objective, Krishna. So it is not that like traditional course running in any yeah, that is actually it is the skill, the degree with the skill. So definitely it will publish your project in a high impact journal. Yeah, definitely. Actually, because we have published already few papers from the internship also. So if you opt this course, so definitely you will get publication at the end of your course with your degree. Okay. And the third thing is that this type of course will run frequently. As I mentioned very beginning, that is to develop your machine learning, you have to learn Python and you have to spend time that is the course. And such kind that you are providing the code, you can use this code to improve your data, improve your model. Okay. Okay, and as Manishka mentioned, that is you will get the feedback form. So you have to fill the feedback form, and then you will get the certificate. Okay, in between, if you have any question, ask. Otherwise, you will go for the final wrap up session. Okay, these are the links. That is our social media link: YouTube, Instagram, LinkedIn, Facebook. So you can follow us, or also you can check our website about our work, our papers, etc. Okay. Okay. Omkar, you can continue.
Yes. So thank you all. So to conclude the workshop, Anirudh will deliver the closing remarks, expressing gratitude to all participants, speakers, and organizers who made this event success. Anirudh, over to you.
Okay. So thank you everyone. On behalf of SilicoScientia, I would like to extend my heartfelt gratitude to each and every one of you for being part of today's hands-on workshop on AI and drug discovery. It has truly been an inspiring and engaging session, and your enthusiasm, curiosity, and active participation made it a great success. We are especially honored and grateful that participants from different parts of the world have joined us today. Your presence reflects the global interest in leveraging artificial intelligence to transform the future of drug discovery, and it has enriched our discussions and collaboration throughout the session. Over the course of the workshop, we explored powerful tools and techniques and real-world applications of AI in drug development pipeline. We hope you gained valuable insights and practical skills that you can carry forward in your own research or professional journey. If you have any questions, need clarification, or would like additional support, please feel free to reach out to us anytime via email. We are always here to help and continue the conversation. We also welcome your feedback and suggestions. If there are specific topics you are passionate about or eager to explore in future workshops, we'd love to hear from you. And please fill the feedback form to receive the certificate of the workshop. For those of you interested in advancing your learning even further, SilicoScientia also offers a comprehensive course on AI in drug discovery designed to deepen your expertise and expand your capabilities in fast-evolving field. So thank you once again for being with us today. Keep learning, keep exploring, and we look forward to welcoming you again in future sessions. Until then, stay curious and stay connected.
Okay, thank you. Okay, so I think Manish already shared the feedback form in chat. So please fill the form and fill the form and whatever the name you want to write in your certificate, please mention clearly. And if anyone wants to say something, please unmute and say about today's workshop and also your genuine feedback. That is what it is really helpful or we need to improve because it's required for us. Okay. Yeah, and so that whatever you felt that is it is poor or it is good, so you have to rate 5 for best, I think so. Yeah. And yeah, Sujit, definitely we'll let you know if anything is there. If you'd like to anyone talk, just unmute yourself and just share your feedback. So, okay. Is it done, everyone? The feedback form? Just tell us so that we can just finish. Okay, so I think we should wrap up. Okay, on just onboard it.
Yes. Okay. So thank you all for joining, and a session or hands-on training and the workshop is ended here.
Okay, thank you. Goodbye and good afternoon to all.